And then we invite people with whom we collaborated on projects, we invite them to come and present the outcome of their projects. So we worked with them and they got nice results and they come and present this. And we pepper in presentations from members of CNAG that are in the area. As you see today, we have single cell, we have spatial, we have long reads. So those are the sort of the technology spaces that we are working in. Now, one big reason why we're actually able to do this symposium is obviously because we're here, but is due to the sponsorship of different companies. And this year, we've had 5 companies: Illumina, Oxford Nanopore Technologies, Bonsai Lab, Nanostring, and Longwood, who have given us financial support to run this, and that pays for the rent of the room, the food that we will have later, the coffee breaks, the travel of the speakers, and so on. We chip in some, but I have to say this year the sponsors have been extremely supportive of what we do here. Good. I'm going to give an introduction, and it's going to be very basic. It's just about what CNAG is and where we come from and what we do. But I'm not going to go into much detail because I leave the detail to the other members of CNAG who will explain in their particular topics what they do, what they have, what we can offer, how we sort of work, and so on. Okay. So by way of introduction, CNAG was created in 2010. As many of you might know, we started off as sort of being hosted by the Parc Científic de Barcelona. This is this particular site here. It's a business park. That was about the first 5 years of our activity. Then we were moved administratively to the CRG. So we were part of the CRG from 2015 until the middle of 2023. And finally, in 2023, actually on the 29th of December, 30th of December last year, David and myself, with a lot of machination from government people and ministers and everything, got the go-ahead to create the legal entity Consortium CNAG. And we did that, and then we spent the first 6 months of this year transitioning the administration from the CRG to ourselves, and now since the 1st of July we are actually handling our own administration. We're still a little bit chaotic, but we're getting more organized and so on. But I think in terms of working with our collaborators, so people who do projects with us, I think probably not a lot has been noticed. That's what I think, because our project managers are the same. The mechanisms of how we work with our collaborators has remained the same. We do the same kind of work. If there is something that has changed and you think it's negative, to the negative, come and talk to me. I will listen very well to what you have to say, because this is really important to us. We can only do our work if the scientific community comes and works with us. And we want, obviously, to satisfy the scientific community. Now, one thing that is really important in what we do is we do not want to position ourselves as just being a sequencing service center. That is not our objective. We want to do more than that. We want to support a researcher in the design of his or her experiment and give advice and say, oh, we think you need to analyze 17 samples. 3 is not enough because your statistics are not going to give you the result that you are looking for. But if you do it with 17, it should be okay. So we will do things like that. Or we will say, your sequencing depth should be like this, or the best technique would be this. So we try to position ourselves as work with you on the design of your experiment. Then we have processes of receiving materials that are standardized. We receive materials in a very organized fashion, barcodes, tubes, sort of a system that you can input all of the information that is relevant for us to work through an experiment. Then we run through sample preparation, quality controlling, sequencing. At that point, we're quite much a sort of a black box. This is you don't hear a lot from us. We tell you in 8 weeks' time, we will give you the result. Then we go through quality controlling of data. We do data analytics. If you asked us to do disease gene identification, we go all the way through that, and we hand you something that we think is a result that you can then actually take and say, okay, now I can do my part of the work into this. I mean, we go to the point even of writing materials and methods for you, and then collaborating with you on producing papers, depositing data into archives and so on. So we do a lot of this very heavy lifting stuff. We try to take off your shoulders and do it because we know how to do that quite well. Okay, so been here since 2010. We're about 120 professionals, mainly very young professionals. We are part of an ICTS. This is a sort of a Spanish infrastructure. And our objective is to carry out projects in genome analysis that lead to significant improvement in people's health and quality of life. And quality of life, you can obviously dial that all the way up to planetary health as well. Our objective has never been to be the cheapest at what we do. We don't try to be the most expensive. We try to be as cheap as we can afford to be. But what we definitely want to be is the best. So we want to be the best at what we do. We want to give you techniques that really help you, that really enable you and really bring it to the front. And we want to give you quality that is better than what just a sequencing service provider will give you and just get said, oh, go away. You have fast queues. You can deal with it from this point. This is just a couple of boiler points. The people who are coming for the tour this evening will get to see some of these facilities. We can sequence on the order of about 100 genomes per 24 hours at 30-fold coverage. We have a bunch of Illumina sequencing instruments. We have Oxford Nanopore sequencing instruments. In the single cell, Holger will surely show some of this. We have Chromium controllers, automated systems. And so we have in our more spatial side, we have a Bruker-Vutara microscope, we have a Nanostring Cosmics, and there's more to come there, and Anna is going to say some about that. And then obviously to deal with all of this, we have quite a big computer standing behind this. This computer is about, I believe, I'm not quite sure. I'm going to, next week, I think I'm going to go and see the new Barcelona supercomputer, and I don't actually know how big it is. It used to be that we had about 20% the capacity of the Barcelona supercomputer, but I don't know what the specification of their new machine is. But, you know, it truly is 10% of what those guys up there have, and they do only compute. We do a couple of other things on top of it as well. Okay, how are we organized? We have basically two big units. One unit that takes care of the laboratory, sequencing unit headed by Marta. We have the bioinformatics unit headed by Sergi, who's back there. And then we have a quality system wrapped around everything we do, and a quality manager who stands outside our general system. We run the entire place under an ISO certification and accreditation. So we have 9001 and 17025. We have a couple of other ISOs that are in preparation, such as the 27001, which we are nearly done with. 27001 is all about data safety, security. It's everything about how do you actually hold your data, what is the risk that someone will break into your computer and take your data away from you, or encrypt your data, or do things like that. This goes all the way down to locking doors in certain places in the building and having controlled access and so on. So, and then very coming up soon probably as well is 15189, which is a clinical accreditation for us to provide exome sequencing or whole genome sequencing or RNA sequencing, and then a few that go around proficiency provision. Here's some numbers of what we do. We work in different areas. We have obviously a lot that hits cancer, a lot of things that hit sort of disease genomics, which is usually a rare disease, functional genomics, RNA-seq, epigenetic analysis going in, and then work on animals, plants, microbial, and a few others. So we have we have quite a wide palette. Essentially, the tools underneath, the instruments, the methods are the cornerstone. So we do things based on the methods and not as much on, we only work on cancer, but we will not look at a rare disease patient because we are a cancer place. So we really go around the technologies, have the technologies really well standardized, and then apply it in the best possible way to the problems that we're trying to solve. Then on top of it, we have our own research activities, and at the moment there are 8 PIs in this space taking care of different bits. I have to apologize. I had to, unfortunately, and I didn't get time to talk to Tyler. I had to sort of leave Tyler out of the speakers. I wanted to put him in, but I ran out of space in the day, and I wanted to have him in, but I just couldn't fit it. Otherwise, you would have had to stay here until 6:30 in the evening, and I don't know whether anyone wanted to do that. So next time it's Tyler's turn when we do this. Maybe we have to do sort of biogenome thing next time we do one of these consortia. The space up the front. And then we have a new team leader that joined us a few months ago, which is Anna Pascual. What are the areas that where we're active? Well, we have 6 main pillars. There's a 7th one that is under construction. We have 6 main areas, obviously personalized medicine around projects like the Spanish Impact project and GA4GH and so on. So we have projects running in that. Rare diseases has been a major topic. We have built a rare disease database system called the RDConnect Genome Phenome Analysis Platform that is used in multiple rare disease projects. Cancer is the reason why we were brought here. So we were brought here to support the Spanish contribution to the International Cancer Genome Consortium. Single cell analysis you will hear a lot about today from Holger and so on. Genome in action, which is sort of a different word for epigenetics. We have a lot of activity in that, and then in the last 3 years or so, we've sort of built an awful lot of activity in biodiversity through the ERGA, the European Reference Genome Atlas, and then EU-funded projects such as the Biogenome Europe, the Catalan Biogenome Project, and ORG1, which we'll hear about later on today as well. Okay, and I think I'll just quickly flash the thing in front of you who is going to be talking. This is going to go very fast. And with that, I'm going to sort of open the scientific part.
So we're running late already. Okay, good morning and very warm welcome also from my side. It's very nice to see so many friends and colleagues that really helped shaping the single-cell landscape of Barcelona over the last years, and I think we can be very proud of what we have achieved putting Barcelona on the cell atlas of the world. And initially, I guess we had been more recognized for the technical expertise as a technology hub, but now we're moving more and more towards the translation and also clinical research context. And my lab, as you see here, kind of focused or followed the same trajectory, initially focusing more on benchmarking to make sure we have protocols in place to generate high-quality data at scale, cost-efficiently. But so following that, we really kind of moved after establishing a framework that allowed us to generate robust data at scale, and also having the computational tools in place that came along, new technologies that were developed and implemented over the last years, really where we are now in a place to develop large-scale projects. And we focus mainly on basic research, which is our contribution to the Human Cell Atlas project, but also we are now more and more moving towards translational and clinical research, and I have some projects to present for that during this talk. In general, what we're doing is using the human as a model system, where we really try to map out the full plasticity of cell states during perturbation. And perturbation in this sense is a disease progression, a treatment response, or the lack of a response. And we kind of really to map out this space entirely, mainly focus on immuno-oncology and more recently inflammatory diseases. Those projects where really immunology is at the heart. And for the scope of this meeting, though, I will more focus on kind of application examples. I will not go much into details of specific diseases or cell types and states, but really give you kind of an overview of applications that can be applied on specific reference atlases that we generate in the context of the Human Cell Atlas, but also now more and more for diseases and even in clinical trials. I will only talk about single-cell projects, and Ana Pascual later today will talk about our spatial activities and the new cool stuff coming up there. As a reminder, so at the core of all data analysis or meaningful data analysis is a high-quality data set, where we then over the last years really try to maximize information content per cell, so through a lot of benchmarking and protocol implementation work, and to minimize batch effects, to minimize biases, to make sure that you really end up with a UMAP, so a two-dimensional representation of single cells, as you see for technology C, and you're not kind of left with a data set of low or inferior quality that really does not allow you to map, for example, rare cell population or more subtle cell types and states. So we've done such exercises for single-cell RNA-seq, published in 2020, but more recently we have repeated the same exercise for single-cell ATAC sequencing, published this year. So for RNA-seq, we came in very early, and we benchmarked around 13 different methods. For ATAC-seq, the landscape was less different. So we had only 4 techniques or 4 technologies available, but in this case, we mapped the different versions and the different kind of protocols that were developed on those technologies, especially for the 10x genomics assays. For example, the mitochondrial single-cell ATAC sequencing protocols. And here, I think what we identified in the end is really that the 10x version 2 of the single-cell ATAC-seq assay showed the lowest quality. Do I have a pointer here? Showed the lowest loss of reads during quality control, so the highest number of uniquely mapped reads, but also the highest information content, which is for ATAC-seq the number of fragments and peaks, so really the information that you get from a single cell about open chromatin sites, and that then allow you to identify differentially associated, differentially accessible regions that then enter as a key metric in the downstream analysis interpretation. So this kind of a wrap-up of our single-cell benchmarking implementation activities, but today's talk really should focus on application that we apply on reference atlases, and here focusing on health on the one side and disease on the other side, focusing on the human cell atlas, human tonsil atlas that we've been generating and that is now accepted in immunity, but also talking about inflammatory diseases and specific use cases of the applications then. So I hope at the end of this talk you're kind of inspired to use those reference maps for your own research and to go beyond the today presented. Therefore, we really try to make those data sets as accessible as possible, the code as accessible as possible, really to accelerate knowledge gain beyond the here presented. Sorry. So the first project I'd like to present is our effort to generate an atlas of the human tonsil, which had been a great collaboration with our partners at IDIBABS. So this had been an ERC synergy grant together with Elias Campo and Iñaki Suberi-Martin. And in total, we've generated tonsil data across 17 different donors, across different age groups. I think the unique feature of this atlas were the different modalities that we put on top, in addition to RNA sequencing, which allowed us to kind of describe a comprehensive composition of tonsils in older and younger individuals, but also then using the additional modalities to dive deeper into mechanisms and into functional states of cells within the human tonsil. So here, in total, we generated more than 500,000 cells across a reference data set that you see here, but throughout the revision, we added additional validation data sets that then, as you can see here on the left, can be kind of annotated at very high level, so the major cell types that form the human tonsil, but then having so many cells available really allowed us then also to dive deeper into specific cell lineages and really to come up with a kind of complete or more comprehensive composition analysis. In the end, diving deep into lineages and describing around 121 cell types and states. So this annotation process had been a very manual process through looking at marker genes together with our next generation immunologist, Juanito, in the group, providing for us really a resource moving forward into the analysis interpretation, but also obviously for future analysis and for the community, a resource for groups working in the area of secondary lymphoid organs. And for me, one of the most important outcomes of this work is actually in the appendix. It's a glossary really describing in details these 121 different cell populations, their marker genes, their multimodal features that really kind of guided our annotation process and hopefully guide then future analysis and follow-up work in the same community. So this is just one example where we performed a deep dive in the CD4 positive T cell compartment. So here in the tonsil, the CD4 cells that start being naive. So we have the light gray cells here, the naive T cells, that then following activation through, for example, dendritic cells, split into two lineages, here the follicular helper lineages, and then the non-follicular helper lineages, differentiating in effector memory cells, regulatory cells, for example, and helper cells. And this is very much driven by two transcription factors, BCL6 for the follicular helper lineage and BLIMP1 for the non-follicular helper lineage. And as we had different modalities available, we could kind of accumulate information and evidence for annotating the distinct cell population that follow the trajectories into the follicular and non-follicular lineages. And one of the first finding and kind of key message from the initial stage of activation is that there's an early bifurcation already in the central memory compartment. So we have the naive cells here, which then, following activation, differentiate to central memory cells. And we see already a kind of a bifurcation and a pre-commitment of these central memory cells to the follicular or non-follicular lineage, then supported by gene expression markers, spatial composition, protein markers, so we have kind of all information available to compile evidence supporting those claims. But so beyond this compositional analysis, we then also used the reference as a reference to map diseased samples on top. First of all, for an automated annotation, so we could directly transfer labels from the Curie dataset, from the reference dataset to the Curie dataset, but also immediately kind of look at composition shifts in a Curie dataset that can come from a disease as compared to the healthy control. And what you can see here, it's a bit unsharp though, apologies for that, is the tumor microenvironment of a mantle cell lymphoma patient. So mantle cell lymphomas rarely manifest also to the tonsil. And here we looked at the tumor microenvironment alone and mapped it to the healthy tonsil atlas reference. So we could transfer the labels, but we could also immediately see a shift of composition as compared to the healthy tonsil with a strong enrichment here in the T regulatory CD4 T cell compartment. So then we've done this exercise a bit more systematically, generating a validation data set. So this is our initially reference as a discovery data set and then a validation data set, which we kind of generated throughout the revision process, which then had almost equal size as the original atlas. And that then allowed us to project the validation on the reference, first of all, to validate our subpopulations and subpopulation markers that you can see here on the right-hand side. But then also we could automatically transfer labels again for the full atlas that you see here on top, also again for the CD4 positive compartment. And then as the validation data set was more biased towards older individuals, we could then look into kind of composition shifts, for example, the age-related reduction of germinal center B cell activity. So kind of following these kind of generation of healthy atlas, we then really ask how to use single-cell sequencing in the clinical context and really to make kind of an impact on a patient and how to kind of transform single-cell sequencing in a clinical tool. for example, for diagnosis as a diagnostic tool, but also to stratify patients and to inform about therapy decisions, for example. But here the problem always was the lower sample cohort size, so the lower number of patients. So in the tonsil we had 17, and this was basically not enough to extract enough features that then would allow us to discriminate healthy from disease or to classify patient into different disease categories. So then for the next project, we went from the 70 patients that we have in the tonsil atlas to much larger numbers. So in total, we analyzed 300 or more than 350 patients, more than 2 million cells before quality control, really to learn a generative model of immune cell states or immune cell plasticity. And now we had really sufficient patients to train classifiers as well. So with this project, we kind of worked on generating in human inflammation atlas of cells in circulation. So here we are looking mainly at PBMCs from patients suffering from inflammatory diseases, focusing on IMIDs, so immune-mediated inflammatory diseases, but also chronic inflammation like cirrhosis, asthma, COPD. And here really the integration of patients and cohorts allowed us to learn, if you wish, a foundation model of inflammation or inflammation model of cells in circulation. And then we paired that with interpretable machine learning, really to kind of classify cells, to extract biomarkers, but then also using the embedded space to project new samples on top and to use that as a sample classifier and potentially as a proof of concept using PBMCs as a diagnostic tool for inflammatory diseases. So as for all large-scale projects, one of the major problems is integration. So on top, you see the unintegrated object. At the bottom, the integrated object. So here we're lucky that all of the data sets came from kind of 10x genomics chemistries, but we were facing different versions, version 1, 2, 3, different protocols, 5', 3', different multiplexing protocols. So really kind of what you see on the top facing batch effects that needed to be removed during data integration. And here we very much aligned our integration strategies with the activities in the human cell atlas and used SCGen, SCVI tools, which has been used already in the lung cell atlas that was published a few months back in Nature Medicine, but is now also the method of choice moving forward for data integration in the immune cell atlas. So this is a kind of the fully integrated object of more than 1,700,000 cells. And on the right again, you see the kind of level 1 high-level annotation, major cell types and states. But as we've done for the tonsil atlas, we then zoomed into the specific lineages and performed a more granular annotation of cell types and states, because those then we wanted kind of to link to specific disease process and inflammatory processes. And this is basically what you can see here as a summary. So initially we used the multivariate linear model to extract kind of general trends or inflammatory states related to specific diseases. So as a disclaimer here, we are not working on the full transcriptome, so it's not highly variable genes, but it's really a pre-selection of genes that were previously related to inflammatory processes that you see here on the, probably see, but you can't read it. So those are inflammatory processes with a predefined set of genes, around 444 genes, that then allowed us to see general trends. So we see most of the samples and diseases shifted to the right-hand side. As we're looking at inflammatory diseases and inflammatory process, this hasn't been a big surprise. But then we kind of zoomed into specific diseases, specific processes. And one of the kind of most striking differences that we've seen is in interferon response, we've seen a high enrichment in SLE patient. This is the blue, I can't probably see it, so you have to trust me. This is the blue here as compared to all the other IMIDs, which are less activated in this specific process related to interferon response. And as we had the full atlas and the full annotation of the atlas, so we could dive deeper than into specific lineages. Interferon activation in SLE patient had been described before in plasma cells or the B-cell lineage, but then we could basically map that activation to the rest of the cell types and states, really providing evidence for a more holistic or systemic activation of interferon response, not only in B cells, but also in other lymphoid populations, CD4, CD8, but also in the myeloid compartment, for example, inflammatory monocytes and dendritic cells. So this was kind of with the linear model, we kind of asked for a gene disease association, but then we moved ahead. We need to look at a more holistically way to extract biomarkers for diseases. And here we teamed up with a group at the EPFL in Switzerland, Pierre van der Geinst lab. I'm probably one of the only persons who can pronounce that name more or less correctly. So this is really an expert group in machine learning, and they kind of came up with a strategy using gradient-boosted decision trees together with SHAP values. And these SHAP values allow you kind of to explain those models, and they allow you to rank genes and cell types by their important contribution to specific diseases. And this is how it looks like on the left-hand side. Basically, you come up with a ranking of genes that you see here on the rows related to specific cell types. In this case, highlighting dendritic cells or monocytes, and then the accumulation here of SHAP values is across all the diseases together. So basically, the message here is that CBER, the gene CBER, is very important in dendritic cells, and then you look at patterns of disease processes that are enriched in those in this relationship, CBER together with dendritic cells or CBER together with monocytes. And I guess that's better displayed here on the right-hand side where we actually see the direction, but we can also kind of zoom into the specific population. So only highlighted here are barrier tissue inflammatory diseases. So on the left, we have skin barrier tissue diseases, and here gut barrier tissue diseases with psoriasis, psoriatic arthritis, Crohn's disease, and ulcerative colitis here. The interesting thing was that we've seen an opposite trend in the gut-related barrier diseases. So we've seen a kind of a gain of functions, an overexpression of CBER gaining importance in the disease process, and on the skin-related, we've seen the opposite. So that there's a reduction of expression related to the to the respective diseases. And then we kind of zoom deeper, obviously, in the function of the gene. I think an interesting observation is, so a bit more about the gene first. So CBER is a super oxide generating oxidase, which basically produces superoxide. Mutations of the gene already have been linked to skin-related barrier function problems, so very much in line what we see in the skin-related autoimmune diseases, potentially hindering the defense against pathogens crossing the skin barrier. But for GI-related autoimmune diseases, we've seen basically the opposite trend, where then the activation, potentially the overexpression or hyperactivity of the oxidase is related to accumulation or higher levels of superoxide in the tissues, leading to local inflammation, tissue damage. In line, there's a knockout model of CBER that when kind of introduced with stimuli to generate colitis. So in the knockout model, there was less disease burden, potentially pointing to the fact that CBER as a gene and related process is very much involved in the kind of disease burden or disease manifestation. So then we basically speculated that CBER and related processes are a potential target then to reduce inflammation in colitis and Crohn's patients. And we went on looking at, so this is all from the blood and cells in circulation, and then we went on validating this in tissues. So this is all single-cell sequencing work together with Azucena Salas from Hospital Clinic and IDIBABS, where we then looked at single-cell data from control patients, Crohn's patients, colitis patients. This is only the myeloid compartment, and what we've seen basically that in tissues in patients we've seen an increased expression of CBER and kind of an increased abundance of these myeloid cell types with CBER hyperactivity. For us, basically pointing to the fact that we can detect local perturbation or local inflammation also more systemically in PBMCs, and that really motivated us to use then PBMCs as a resource for biomarker or for a kind of liquid biomarker resource for more kind of patient-related diagnostic or treatment monitoring processes. So here, in the last kind of part of this analysis, we then really ask if we can use single-cell sequencing as a diagnostic tool for inflammatory diseases, and therefore we use the generative model that we generated during integration of the data, and now projected new samples on this embedding space for classification. So here, to reduce noise, we basically used a meta-cell approach per cell type and state, which really then allowed us also to see where the classification accuracy comes from overall, but then also in cell type specifically. And I hope this is now. Okay, so, and basically what we've seen is an amazing classification accuracy of greater than 0.9%. So really pointing to the fact that blood has enough information to be used as a classification tool, and single cell providing kind of the resolution to zoom into specific cell types, but then as an aggregate. So what you see here in gray is the aggregate across all cell type lineages, greater than 0.9. So this, I think, is an amazing result. But even looking at the single cell types, you see very high accuracy, pointing to the fact that most of the cell types contain sufficient information to classify patients. There was one significant outlier here with the HIV patient, where there was very low accuracy in the myeloid compartment, very high in the lymphoid compartment. I'm running out of time. Last slide. Really pointing to the fact that it can be related to the biology of the virus infecting CD4 positive cells, but really motivating us also using this holistic approach, looking at cell types in general. So now as a follow-up, I think we're very motivated using these kind of high predictive power of single cells in diseases. We're moving now to larger data sets, more patients, more diseases. Inflammation, Atlas version 2. So stay tuned, there's more coming. And with this, I have to end. And I thank my wonderful group. So big disclaimer, I haven't done any of this. It's all work of people within the group. Very big thank you to the Sinak for creating such an amazing culture and atmosphere of kind of supportive and creative science. This was us a few years back, so I thank my current members, my former members. So we are much more serious now. And thank you for your attention.
So first of all, thank you very much to the organizers, of course, for inviting me. It's a great pleasure to be able to show you actually a project that's been a collaboration with Holger, so it's very nice to just follow up on what he's saying. And I don't know how to get to my slides, because these are Holger's slides. Yeah, so continue. I've got some backup slides as well. We'll have this problem. You have a lot of backup. There we go. This is me. All right, so I'm going to tell you a little bit, basically more about the biology that we're trying to study with all of these technologies that Holger has just explained to you about. And what we work in the pediatric hospital here is in childhood cancer. And the first thing that I always like to point out is that childhood cancer is actually a very, very different disease from adult cancer. And that kind of stands to reason, because children are not just really small adults, they're growing, they have a high metabolism, they're still undergoing development. But there's another three features that I would like to highlight, which are, I think, very important for what I'm going to tell you. So first of all, childhood cancer so in adult cancer, if you look at the incidence, basically the older we get, the more likely we are to get cancer. So it's a disease of aging, because you accumulate mutations. In childhood cancer, it's very, very different. So you have a very big peak, so very narrow diagnostic window, so a very specific age where you diagnose this cancer. So in this case, for DIPG, for example, it's around 5 years of age. And then when you become older, you're basically never going to be diagnosed with these cancers. And the reason behind that is that people think that this narrow oncogenic diagnostic window is linked to a narrow oncogenic window, so a very narrow window during development that goes wrong. So we think of childhood cancer essentially as development that has somehow gone wrong. So on the other hand, a further difference is that the driver mutations are very different, so we don't see BRAF mutated or KRAS or any of those classical cancer driver genes. We actually see epigenomic mutations, so problems in the epigenomic reorganization, which kind of also goes back to development, because in development you have such epigenomic remodeling, and when this goes wrong, this is when you start becoming developmental cancer. And lastly, of course, in childhood, you don't have all the time to accumulate the mutations, so these tumors have a very low mutational burden, which makes them relatively homogeneous. And here comes the problem, that we, especially when we talk about solid tumors, we don't really know how to treat them. We don't have personalized therapies. So what we do is we use radiotherapy and chemotherapy, and both of those are both toxic and immunogenic, so basically generating a much more difficult disease to treat, and on top of that have very, very high and long-term side effects for the patients. So essentially what we want to do in the lab is kind of understand the biology that underlies childhood cancer to be able to get to precision medicine. This is the big idea. And we argue that we need to understand the disease to really be able to target it properly. But of course, there's some very important challenges. So first of all, these are very rare tumors, luckily, but in terms of analyzing and understanding them, this is a problem. Second, as I already told you, they arise in prenatal development, which is a process that we still don't fully understand. And not only it's very complex, but it's also characterized by very fleeting cell states, and the context changes continuously. So it's very difficult to then pinpoint this very specific oncogenic window that I've been talking about. So in the lab, we work on several different tumors. So we work on Ewing sarcoma, osteosarcoma, neuroblastoma, and here my slides have somehow moved. But anyway, also on malignant rhabdoid tumors, which is what I'm exclusively going to talk about today. So these malignant rhabdoid tumors, many people call them kind of the epitome of epigenetically driven tumorigenesis. And this is because essentially they follow all of these three kind of very key features of developmental tumors. They have a very narrow oncogenic window. They're diagnosed usually before 2 years of age. So there's many children that are just born with these tumors. So infants, we're talking really about infants. The mutation burden is very low. Here, this was a study in Nature some years back that compared adult cancer mutation rate to so pediatric cancer mutation rates to adult cancer, and there's like an order of magnitude drop. And the rhabdoid tumors are here in the low range, so below 0.1 coding mutations per megabase. So this is really, really low. And just to note that all of the ones that are lower are actually not very malignant. They're quite benign tumors. So this is a very malignant tumor that has a very low mutational burden. And the driver mutations, and the only recurrent mutation that we see, the only recurrent mutations we see in these tumors are in the Swicenef chromatin remodeling complex. So here you have the rhabdoid tumors. More than 95% of them are mutated for a gene that's called SMARCB1, and this is a core constituent of the Swicenef chromatin remodeling complex, highlighting that, again, this is the problem that drives this tumor is epigenomic remodeling. 5-year survival of these patients, remember they get diagnosed when they're around 2 years old. The 5-year survival is less than 20%. So we really need kind of urgent precision medicine. One thing that I wanted to highlight is that these tumors clinically are actually quite heterogeneous. Histologically and genomically, they're very homogeneous, but clinically they can arise in many different parts of the body. They can arise in the central nervous system, in the kidney, in the liver, in soft tissues, and so on. And there's been a huge controversy related to these different localizations about where these tumors come from, right? So there's many different papers that have tried to figure out what the origin is and where they come from, and it's been extremely difficult, and there have been propositions of neural stem cells, general mesenchymal stem cells, neural crest, but nobody has been able to fully prove it in a kind of fully accepted way. So what we do in the lab, well, it's a difficult question, so we try and address it from many different angles. So on the one hand, we kind of characterize rhabdoid tumor patient samples, and this is what I'm going to speak about today. And on the other hand, we also try and generate faithful models of the origins of these tumors, with the idea ultimately to get to personalized therapy, which could take many different phases. It could be epigenomic therapy, it could be differentiation therapy, gene therapy, stromal targeting in the form of immunotherapy, for example. So as I said, I'm going to talk about the characterization because this is what we've been doing in very, very close collaboration with the Sinak, with Holger and Ines, a postdoc in this group, which has been actually it's been a really fun collaboration. And here we're kind of at the intersection of cancer and rare disease because, as I told you, our cohorts are very, very small. So we're lucky enough to have a biobank cohort of 20 patients that we've been able to select, but 20 for us, this is a huge number. I know for many cancer types, this is really minuscule. But 20 for us, this took this reference hospital in Catalonia more than 15 years to collect, to have enough tissues to do this. And basically what we're doing is, what we did is single nucleus multiome combined with Visium spatial transcriptomics, and the idea at the very beginning was to annotate this with published data sets, and then we would have like an idea about the intra-tumor heterogeneity and the heterogeneity between tumors as well. And hopefully from these transcriptional states that we would identify, maybe we would be able to get to like, to identify the cell of origin, because there's also an increasing number of atlases during development, during human development that have been published. So because there's so few samples, so first I'm going to give you like a little bit of a case study of how this looks, because the spatial transcriptomics data is actually really revealing about the type of things that we see. So this is one case. This is an extrarenal rhabdoid tumor. This is the single-cell transcriptome, where we actually already identify a heterogeneous transcriptional profile. So despite the fact that they have a very simple genome transcriptionally, we already identify many differences. And these differences, actually, we see that they're distributed in space. And then thanks to this algorithm that Holger's group developed, which is Spotlight, we've been trying to really deconvolute in space, kind of in an almost single-cell resolution way, to see what the transcriptional profiles are in space, in space distribution. And on top of that, so basically we're interested to see how the signaling goes and how these different transcriptional profiles are, why they're distributed in space. And here I'm showing you PTPRC. This is CD45, so this is kind of an immune fraction of a tumor. Also, you see this with a CD68, so macrophages are one of the dominant immune cells in these tumors. And what we saw, what I found particularly interesting, is that you have like a very intensely different transcriptional profile just immediately next to these immune cells. Then they're characterized by FGFR1, and we thought that, okay, this might be fibroblast, but actually when you plot a fibroblast score, you see that the fibroblasts are much more around the periphery of the tumor. So then we thought, okay, so could it be that actually the tumor cells are taking on this function, that they're changing the transcriptional profile immediately adjacent to the, to the immune infiltrate? And then basically this is what we looked at. So this is the core of the tumor, and remember, SMARCB1 loss is what characterizes the tumor cells. So the stained cells are actually the stromal cells, and in tumor core you see that you have very big nuclei of the tumor. And you start going to the periphery where you have more stroma and these kind of maybe more immunofibroblastic cells, and you see that already just looking at the nucleus, the morphology, so the phenotype of the tumor cells which lack this SMARCB1, this already changes, and this is without any changes in the kind of genome of these cells. So they appear to have very high plasticity. And the cool thing is actually that we identify these cells. This is one of the particular cell populations that we identified as tumor cells in the single cell profile of like in the clustering of these cells. And we can see that, okay, the clustering actually relates to very specific interactions within the tissue, so to the interaction with the immune cells. And then we can try and start teasing out what the specific signaling events are that happen in the tissue. So one of the things that I want to highlight here is that these tumors have an extremely highly plastic transcriptional profile, and this is something that we also see when we look at the combination of tumors. So this was a case study, and now I'm going to show you what all of the tumors that we have sequenced and integrated so far look like, and they don't integrate at all. So it seems like every patient goes on its own, more or less, right? There are some that are more similar to each other, but they're very, very independent. And let me remind you, this is actually not very surprising if you think about it, because these are the tumors, the locations of the tumors that we sequenced. So every tumor that we have has a very, very specific niche. So if we have within one tumor next to the microenvironment, so next to the immune infiltrate, you already have a different transcriptional profile. It makes sense that if the tumor is in a completely different niche, then it will have also a different transcriptional profile. So this again highlights that there's a very, very high degree of plasticity. But again, then we also wanted to see, okay, there's a high degree of plasticity, but how can we really take information from this? How can we figure it out? So there Ines did kind of a PCA analysis trying to figure out if there are any big differences between the different groups of tumors that we have. And indeed, we saw that, for example, the intracranial tumors are much more different from the extracranial ones. Now, we don't know if this is because the brain is very different, so the niche is very different from anything that is outside of the body, or if this might point to difference in the origin of these. And then, of course, we can also look at the loadings. So Ines can look at the loadings, and we can try and figure out whether there could be like new intervention strategies that we could utilize specifically for subgroups of these tumors. But this is something that is very preliminary and we have not been able to explore quite yet. So again, I told you that the cell-cell interactions within the tumor matter a lot. So can we actually exploit this? Can we find specific patterns that hold through for all of the tumors? And this is an analysis that we did where Ines looked at patterns of co-expression genes based on their spatial variability. So basically, she identified these genes that generally tend to be co-expressed together. And she did this for all of the different tumors that we have. And then she did a hierarchical clustering trying to figure out what is similar and what is different. And here you see that we do have a cluster for the tumor, so for the regions that presumably interact with immune cells. And this could be really, really important for us to look at because the immune cell infiltration of these tumors could have a very important impact on the treatment of the patients. And why do I say this? Well, the rhabdoid tumors, this I showed you already, but there's also a very big difference in the amount of immune infiltration that we see, depending on the different subtypes. And we see this different immune infiltration also in the specific tumors that we sequence. So we range anything from 50% immune or stromal infiltration to something that is clearly less than 10. So there are very, very big differences. And if we have high immune infiltration, maybe we can exploit this therapeutically. So we're very interested to know what drives these differences because it's not mutational burden. So we're interested and we have some ideas about this. But what I'm going to show you now is why it's important to understand it, because maybe these tumors could actually benefit from immunotherapy. And there I'm going to go into like a very specific case that we have recently sequenced of a patient. So here I'm showing you the MRI of a kidney rhabdoid tumor patient at diagnosis. This is a little girl that was 5.5 months old at diagnosis and had this massive tumor mass. So this is the frontal view, and this is a cross-section. So you see how much this tumor filled actually the abdomen of this baby. And basically what happened, there was a full nephrectomy. The tumor was removed. And then because we don't have specific treatments, this is the treatment schedule that these kids undergo. So this is 10 cycles of chemotherapy plus radiotherapy, and you add all kinds of chemotherapy, right? You have vincristine, doxorubicin, cyclophosphamide, carboplatin. There's a bit of everything, right? In the hope that something will work. And in the hospital, so what we do for these cases that are, the prognosis is so bad, there's a program where you look in the histological sections of the patients, you look for something that might give you like indication of what new or additional treatment approach might work. And basically what they found, and here I'm showing you two different locations of the tumor. So here you see SMARCB1, so SMARCB1 is lacking in the tumor cells. And we see that this particular tumor was actually very highly immunely infiltrated, not uniformly throughout the tumor, but it had considerably high levels of CD3 infiltration and also high levels of PD-L1 expression, which of course opens up to the opportunity of giving anti-PD-L1 antibodies. So indeed, after the first cycle, apart from the second cycle onwards, we were able to give this girl atezolizumab, which is the anti-PD-L1 antibody. And then at the same time, we were, with every cycle of chemotherapy, taking blood. And by now, we have been able to do the single-cell RNA sequencing and TCR sequencing of 3 of these time points to kind of see if we see any response and how the immune environment of this girl is responding to therapy. And so this is 10x sequencing of the peripheral blood, so PBMCs of the girl, and this is what this looks like. So you have a big compartment of NK cells, and here kind of the different tumor-like cells. This is of all of the cells that we sequence together, but of course then if we separate them across the 3 different time points, so this is the time point before we added immunotherapy, and then you see that after therapy, really the landscape changes a lot. So we see a big shift towards NK cells, after the 2 different time points, but also we see an expansion in what here you see are the CD8 pre-exhausted cells, so these are presumably active in immunotherapy, and also the proliferative T cells, which you see here. So from this indication, it kind of looks like maybe this therapy is working, which is very cool. And then from the TCR sequencing also, we could also have a look at the clonotypes, so basically at the diversity of the different TCR clones that we see in the blood of this girl. So before immunotherapy, of course, as expected, there was a high number of clonotypes which decreased with treatment, which basically means that hopefully clones that are active against the tumor are expanding. And indeed here, just by plotting the size of the clones against this single cell map again, you see that the hyper-expanded clones map to this population that is the pre-exhausted. So either the proliferative or pre-exhausted T cells, which again indicates that probably these are working quite well. And just to say that from the very deep TCR sequencing that was done, we can also have an indication that these clonotypes that we see were probably already present there before treatment, and this is something that then was expanded, possibly, we cannot be sure, but possibly due to the immune checkpoint blockade. So where are we with this patient? So this is what I showed you already before, the MRI diagnosis. This is after 4 cycles of chemotherapy. She was in complete remission, so it looks like the immunotherapy is up till now working well. There have been a couple of other follow-ups with ultrasound, and we're pending the next MRI, so we're really crossing our fingers that she continues to be in full remission, kind of hoping that this could be like an avenue that could help at least some of these patients. So what I want to highlight is that all of these technologies really give us the opportunity to have a real-time follow-up of treatment response, and also potentially predict a relapse if we keep following in the blood throughout time. And this brings me to the end of my talk. I just want to highlight a couple of take-home messages. So first of all, these tumors are very rare and they're very genomically simple, but despite of this genomic plasticity, we see quite a great deal of transcriptional plasticity. And there are some core transcriptional features that separate the tumors into kind of the extracranial and the intracranial group, and this might be something that might be useful for using different treatment opportunities for these different modalities. And as I showed you, some rhabdoid tumors are very highly infiltrated, and specifically the infiltrated tumors, they might be really good candidates for immunotherapy, and this is something that we will keep working towards. And the technologies also give us the opportunity for real-time monitoring of the response of the patient, and also basically giving us an idea of how we should change possibly treatment in the future for these patients. And this just brings me to thank, well, first of all, this has really messed up my slides, but most importantly, Holger and Ines, who've essentially been, have done all of the analysis of this data, and it's been a really fun collaboration. And then thank you for your attention. Happy to take questions if you have any.
Thank you very much. Questions? Yes, here. I think it's being streamed to a couple of people from CNAG who are watching it, so they will not hear if you don't speak into the microphones.
Thanks, Alex. When you say that you find transcriptional plasticity, what do you mean exactly? That you have quite a bit of diverse transcriptional programs going on?
Yeah.
Or that these programs can adapt to some stimuli? Because plasticity to me means this potential of adaptation, but what you seem to observe is diversity rather than plasticity. So but I want to know your opinion about this.
You're right. So from what I show you, what we can observe is that there's diversity of transcriptional profiles, and we can kind of imply that this is because of interaction with the environment. What we see with, we also grow all these tumor cell lines in vivo. We have reconstituted SMARCB1 expression. We're doing many experiments in vitro. And I can tell you that basically, depending on the conditions you grow them in, they look completely different. So the same cell line, when you put it in different conditions, can grow in a neurosphere, can grow attached, can grow elongated as a mesenchymal stem cell. And we believe that this is because we think the origin of these tumors lies during gastrulation. So it's very, very early before during the kind of specification of the germ layers. And basically, they have the potential to really become many different things. So from the cell lines, depending on the way you culture them, you can generate almost anything. And I think this is kind of what we see a shade of in the tumors as well, depending on the environment, the microenvironment that they have.
Any other questions? So I had a question with the tissue, say the stromal tissue that is around the tumor. Do you see any that you didn't sequence in the single-cell experiment, or do you sequence that as well?
So in the very first batch, we didn't do matched serial sections of the single cell and the spatial transcriptomics, and then in those cases it's difficult to say. But then in the later batches, we did actually the serial sections of these two things. So it's kind of
But do you see like a hijacking of biochemistry from those cells by the tumor?
Biochemistry, what do you mean?
Is it taking any nutrients from the environment and using it that it doesn't make it, that the cancer cell itself is not able to make? So does it benefit from its environment? That's my question.
From the data we have, we cannot say that. So I think we have indications that they're interacting, but I cannot be sure that they actually whether the environment. I mean, it seems that the immune environment is kind of skewed towards tumor promoting.
The immune environment is that's doing whatever it does. But what I'm interested in is just the constitutive environment.
So the let's say the kidney.
The kidney, is the kidney kidney cell contributing to, because there must be atlases of kidney or atlases of brain that you can look at what a normal cell looks like, and then you look at the cell there and you say, is it doing the same thing, or is it?
So we're limited in the samples that we have, because basically from the biobank, the samples that were archived are selected for tumor material. Okay. So basically the normal material is not as standard archived. So we can screen to see if there's some pieces that contain some of the normal tissues, but in the first instance we just sequence the tumor. So we don't have this information, although I think it would be interesting to expand the studies and try and have this specific interaction with the resident tissue. I think this would be very interesting.
Any other questions?
I'm going to skip my backup slides.
Okay. You're doing a good job of the next person. Thanks very much. Thank you. The next speaker, who is Susan Klut.
Excellent.
So I was practicing that one for 25 minutes. Okay, good. I'll leave it to you.
Thank you. So, good morning, everyone. I'm very excited to be here to tell you about our experience with the Zenium platform. So, I was invited by 10x Genomics and Bonsai Labs, which I'd like to disclose they paid for my travel expenses today, but all the opinions I'll show you are my own. So first, for those of you who are unfamiliar with the Zenium platform, I have a quick one-slide overview here. So this is the in situ spatial transcriptomics platform from 10x Genomics, and it's based on padlock probe chemistry, which I don't have time to go into today, but I know you all have a lot of info in the tote bag you received, so you can read more about it later if you're interested. And what I've shown here, oh, I'm one slide behind. Super sorry for that. So, why our lab was really interested in the Zenium was that it had a very large capture area, so we could place any kind of sample we could think of, and that's really important as a core lab because you never know what samples you're going to get. And the Zenium also offered a relatively quick turnaround time, so we can run samples in 2 to 3 days, and that meant that we could keep the throughput very high, and this would allow us to support as many researchers as possible at the LUMC. So our experience so far has been really positive with the Zenium. First things that we've learned is that the slide is rather expensive, so it's important to practice. So we encourage all of our groups to actually draw out on a slide what does the capture area look like, and to go to a non-experimental block to make sure that when the big day comes for the real block, they're ready to go. And one other limiting factor is that the slides run with fiducials, and it's important that you don't cover the fiducials that are located here on the top and the bottom of the slide. And I say this because we already had 2 groups who did cover the fiducial, and that means we have to throw away the slide and start over again. Now, my technicians in the lab have told me that the workflow is really easy to use, and about 75% of the workflow is kitted out. So for any of you who've worked with 10x Genomics products in the past, you know this means that it comes with really clearly labeled reagents. You get a nice user guide that has clear instructions about how everything works. And it takes roughly 3 to 4 days of hands-on time. So that looks like shown here on the bottom, where on day 1 we do the sectioning, and the Zenium works with both fresh frozen and FFPE material. So on day 1 you section and then you dry out your slides. Then on day 2 the users usually come to us and we perform the fixation and permeabilization step if they're submitting fresh frozen material, or the deparaffinization and decross-linking steps for FFPE material. which takes us a few hours. And then this is followed by overnight probe hybridization, ligation, and rolling circle amplification, and then we have another stopping point before we go to the instrument. So all of this part here is totally kitted out. And then we have just a few quick steps to go before we're actually able to run the instrument. So the first is to load all the reagents, and this is actually a picture of the touchscreen from our Zenium platform, where it checks to see, did you put everything in the right spot? Everything's really clearly labeled, and it takes you less than 15 minutes to actually set this up. And here's a photo of the inside of our Zenium, where you can see the reagent bottles that go on the left. In the middle, you have some plates with reagents, as well as tips. And on the right, we're able to run 2 slides at a time, so those are shown here on the deck, ready to go. So there's just one more step before the run really gets started, and that's to perform a DAPI stain of the regions of interest. And here I've taken an image from the 10x Genomics site where you can see 2 different slides containing mouse brain hemispheres. And the DAPI stain imaging takes about an hour, and then when you're done, you get a really nice image of what's present on your slide. And all you have to do is go to the touchscreen and select your region of interest. So you can say, just give me everything. If you want to speed up the runtime a bit, you just go with the touchscreen and you choose the regions that contain tissue, and then you're ready to go. And then you get what I like to call the final countdown. So for the next 2 days while the instrument's running, this is all you're going to see on the screen. It's just this countdown timer to 0, right? So as I mentioned, it takes roughly 2 days per run, depending on the size of the panel. But Zenium's working really hard while it's running, and actually, as soon as the run is finished, you've got data that's ready to go. So you get a data file that's been processed on the server that comes with the instrument, and this is ready to be used with the 10x Genomics Zenium Explorer or any third-party tool that you like. And what's really interesting for me as the head of a core facility is that I get a very useful summary file, and this is similar to the web summary outputs if you've worked with any 10x Genomics products in the past, where you can see in one really quick overview what were the key metrics for my run. So how was performance? How was the background probe binding? How does the segmentation look? And within 1-2 minutes, I can see, hey, this was a successful run in terms of metrics, or it was not. Now, in the second part of my talk, I'd like to highlight some of the data that we've been generating with our first few runs. So we've just had the platform since the end of June. So we're still getting started, but we've already run quite a few different tissue types that I'd like to highlight. So the first run that we did was with human Huntington disease brains. These are from the group of Willeke van Roon-Moom at the LUMC, and she's in our human genetics department. And Willeke submitted fresh frozen cortical and striatal blocks. And for this, we used the pre-designed panel from 10x Genomics for the human brain, which contains 266 targets. Next samples that we ran were from human colorectal cancer. These are from the group of Noel de Miranda in our pathology department. And Noel's group submitted tissue microarrays from colorectal cancer, where he's fit up to 40 different punches per slide. And for this, he worked with a custom panel, and we were very happy to get early access to the large 480 gene custom panel from 10x Genomics, which is now a part of their product catalog. We also worked with some human embryo samples. These came from the group of Susana Shuvra Souza Lopes in our anatomy department. And Susana submitted embryoid testes and ovary samples on one slide, and on the other slide, we did a test run with agarose-embedded embryoid bodies. And for this, we used the pre-designed human multi-tissue and cancer panel from 10x Genomics, which contains 377 genes. And finally, we also ran samples from an external user who submitted head and neck squamous cell carcinoma, as well as breast cancer samples. And for these, we used the human lung panel and the breast cancer panel, both pre-designed panels, which contain 289 and 280 genes, respectively. Now, I'd just like to highlight some of the basic run metrics that we got from our internal LUMC samples. So from the Huntington brain, we had the cortical and striatal block, and here we were capturing more than 200,000 cells per slide. And we see a median number of transcripts per cell of 33 from the cortical block and 45 from the striatal block, corresponding to a median number of genes per cell of 24 and 30, respectively. Now, for the colorectal cancer sample, the numbers get a bit better. So we had quite a few cells. We used the space very efficiently here, so more than 700,000 on one tissue microarray and 463,000 on the other. And both of these perform really similarly with 126 transcripts detected per cell, corresponding to more than 50 genes per cell. So keep in mind here we have the very large custom panel, so you can see that we're detecting quite a few more transcripts and genes per cell, which was to be expected. And finally, for the human embryo, testes, and ovary sample, we also had 894,000 cells with median number of transcripts per cell of 34 and genes per cell at 23. And for the embryonic body, cell number is low, but this was sort of a test run for us to see if it was possible to use agarose embedding. So things were very sparsely populated, but we had a very high number of transcripts per cell with one of these standard panels at 85 and 39 genes per cell. Now, the last data I'd like to show is actually zooming into this colorectal cancer tissue microarray. So here on the left is an image of the DAPI stain. So as I mentioned, you can use the space really efficiently with these tissue microarrays. So we have more than 40 punches here on just one slide. And with the DAPI stain, you can already see a lot of nice structures that are present in the tissue. Now, if we take the standard clustering from the Zenium, so not doing anything in a third-party tool, actually, is it possible to dim the lights in the back? Maybe it'll help people see a bit. If not, just use your imagination. So what we can see a bit already with the clustering, you can start to recreate the structures based on the DAPI, and then that's really clear when we actually merge the two, where you can see cell types that are tracking really nicely with these tubules and striatal sections that we see here in the tissue. Now, if we take a look at 4 of the 480 genes that were on our panel, here I've highlighted some members of the integrin family. And what I'd first like to highlight is the really nice dynamic range that we're seeing, where in the third panel we have the integrin subunit alpha E, where we had just 12,000 transcripts detected across all the samples on our slide, but we still see really nice colocalized staining even with this low abundant transcript. Then on the left we have 2 genes with sort of a medium level of expression, and on the right we have integrin subunit beta 1, which was actually one of the most highly expressed genes in the panel, where more than 3 million transcripts were detected across all tissues. And I forgot to mention, sorry, blue is low and yellow is high. But you can see, yeah, very high expression, but still colocalizing really well with what we could see in the DAPI. And I want to zoom in on one of these particular tissue punches. It's from the bottom left corner here, where here I've got a larger view of the DAPI stain, where again you can see there's a lot of structure present in this particular cancer sample. And if we take a look at the clustering, at least what I see by eye, again, this is not very scientific. You saw great clustering analysis earlier. This is just default, what do we find? We see, for me, a lot of pink and red, and then another section of like yellow and green cell types. And what we noticed just in the initial screening analysis is that this integrin alpha subunit is really colocalizing well with this red and pink population, whereas this other integrin subunit alpha 4 is colocalizing very well with the green and the yellow. So, of course, this is just 2 genes out of 480, and we still have a long road to go on the analysis, but this is something you're just really easily able to pick up by eye with the default clustering. So we were very encouraged by these results. So I just want to briefly conclude by mentioning that we found the Zenium to be very easy to use in the lab, so my team is super happy with how the workflows are going. I also find this workflow to be very fast, not just in the runtime itself, but the fact that we have users bring slides to us on Monday or Tuesday, and by the following Monday, we're already sharing data. So I don't know how fast you guys get your sequencing data back for single cell, but I never have data back this fast. So for me, it's really powerful to go from sample to insight, within a week. And finally, the run metrics that we've seen so far from our FFPE materials are looking really great, and we hope to continue to see this level of performance for a long time to come. So with that, I'd like to briefly acknowledge my team at the Leiden Genome Technology Center. So Emil, Yafuz, and Natalia are the ones who generated all the run data I've shown so far. I'd like to again acknowledge our LUMC collaborators, Villika, Noel, and Susanna, for allowing me to present some of their preliminary data today. And I'd like to acknowledge our team at 10x Genomics, who've been very instrumental in helping us get the Zenium up and running. And with that, I'll be happy to take any questions.
Thank you very much. Questions? So maybe you said this. How long do you have this instrument already?
Since the end of June, and we had training in July, which is the worst time to get an instrument because everyone's on holiday. So we really started running only in September, yeah. Anna had a question.
Yes.
First question was, have you, or what is your, let's say, experience when comparing data on the Zenium, fresh frozen versus FFPE tissues? Have you, do you have any insights on that?
So the only fresh frozen run we've done so far was the first one I showed on the human brain.
Yeah.
And brain is tricky, especially a Huntington disease brain. So to be very honest, the metrics we had there were not great. I'm not convinced the segmentation worked as well as with the FFPE. But yeah, it's a tricky tissue. So I've got my next fresh frozen run coming in the beginning of January. That will be with muscle. That will also come with interesting challenges. So yeah, fresh frozen, jury's still out, let's say. But FFPE, I have another 3 runs that aren't shown here, and all of those are looking really quite consistent when we take a quick look at the segmentation. Yeah.
And may I? Yeah. And from whatever the size of the panel is that you are using, you show different sizes, right? Do you have any idea on how many of these unique transcripts do you detect across the tissue that you are profiling?
So let's say I have a panel of 300. Do I find all 300
Yes.
back? Yeah, we've taken the closest look at this with the large custom panel with the 480 genes. There we found, I would say, about a dozen that we didn't pick up. And then it's not clear, is it really just not there in these tissues? Is it something about the probe design? For the rest, we see some level of low expression, but whether that's background or true signal, yeah, they're still working on it. But for the most part, if you scroll through, you see events, yeah.
And then maybe the last question, which is the size of the data that you get out of one of these tissue sections that you've shown?
So per slide, we get 40 to 50 gigabases of process data. You can go back into the raw data if you want. That's quite a bit larger. I would say, yeah, then you're getting close to a terabyte. But in terms of export for us, what we share with the users, yeah, 40 to 50, so 100, 120 gigs per run.
But
Yeah.
you have to store the raw data, right?
Yeah, that's up to the groups to decide how
Okay,
much,
yeah,
some are happy with processed, a lot actually want to have the raw data so they can go back later and redo segmentation or whatever it happens to be. But yeah, it's still quite a lot of data in the end, yeah,
All right, thank you.
yeah.
Thanks.
Why do we need spatial data? Why the spatial dimension on biomedical research is so important? And it's mainly because the organization of biological structures at all levels actually determines their function. And only by understanding the spatial fingerprints at all these levels is where we can actually get a better understanding on mechanisms of health and disease. And actually, the importance of the spatial information is not something that we are now realizing. So histopathology is still now one of the gold standards to diagnose disease. And I'm coming from an imaging lab. The problem is, though, that with H&E or immunofluorescence, for example, you are relying on, at the best, 4 to 5 markers that you can capture from one single tissue section, right? And that, of course, limits the throughput of the analysis that you can perform with that. So actually, what we are seeing now is the leveraging of the technical developments that have been ongoing in the last 10 years in the genomics field, applied to actually the analysis of tissues, right? And however, it's only now that we can combine the analysis of whole genome or whole transcriptome of any of the single cells that we can see in tissues with their spatial coordinates. And that's important because when we look at tissues, we actually can see how there are cells that hang around very close one to the other in particular areas, but not in other areas. So we need to understand how these cells communicate with each other and how the gene expression is actually spatially regulated and why. And that's actually when the spatial tissue profiling techniques come into play. And it actually all started, or mainly started, with antibody-based cycling imaging technologies that were nevertheless of low plexity. And in the last 4 years, let's say, the whole field has evolved a lot, mainly based on the detection of transcripts from tissues. And I mean, it's probably worth also mentioning that There are more than 50 different technologies that allow for spatial tissue profiling right now, that the majority of them have been actually developed in research labs, and that already 10 or more than 10 are being commercialized instruments right now. So this speaks for the interest that these technologies are actually generating in the research community. However, we have a high disparity in the number of markers that we can detect in each of these different techniques, and also very variable resolutions that we get from them. And also, it's also worth to mention that we'll probably be seeing quite a lot of development in the detection of DNA at the spatial level, because this is really not yet as the state is when we detect RNA or proteins. But if we focus on spatial transcriptomics, and we agree that the aim should be to actually be able to capture single cells from any given tissue in the human body, and to do that with single cell resolution, we have to distinguish in between two types of spatial transcriptomics technique. On the one hand, we have capture-based methods that allow us to get transcriptome-wide data at the expense of resolution mainly. And what we get here is, let's say, mini bulk measurements that we can then deconvolute to get into the single cell resolution. On the other hand, we have imaging-based platforms that rely on pre-designed targeted panels. Those panels can be larger or smaller, but the good thing about those is that you actually get subcellular resolution, and you can directly map the transcripts to the cells. Of course, after computational methods had been used to actually segment those cells from the native tissues, right? But why do we want these techniques? Again, we want to use these techniques to actually understand tissue pathology. And this is by acknowledging the fact that we have molecules and cells closely interacting within tissues, within tissue niches, and that those interactions are actually supporting the homeostatic function of the tissues or promoting the activation of these tissues' anti-inflammatory conditions, and how this can lead to disease. And to actually try to fully grasp the whole complexity of these cellular and molecular interdependencies in space is where we can actually use spatial genomics techniques and actually combine those also with classical histopathology analysis. And that's just one example of a study that I did on my previous lab in Berlin, where we actually combined histopathology with spatial transcriptomics and with a cyclic technique for immunofluorescence. And we could actually identify that there are distinct tissue niches in the lungs that are key orchestrators of prolonged severe COVID-19. And so we used expert annotations, let's say, to identify particular regions that were undergoing disease or tissue transformation during disease progression. And we used the Zenium, which is not single cell resolved, but still would allow us to actually detect aberrant expression levels of particular signals in the tissue in those particular tissue areas. And also, since we had the whole transcriptome from those samples, we could really characterize in depth the distinct tissue microenvironments that were found in those areas and the nearby areas. And to actually get to the single cell resolution, what we did is to combine those spatial transcriptomics analysis with this immunofluorescence panel that would allow us to then actually pinpoint which were the cells precisely within those areas responsible for this aberrant expression of some chemokines, and the cells that were responding to that signal as well. And we can actually also leverage the fact that from the same samples we can get single cell or single nuclear RNA-seq data, and we can then deconvolute the single or the Zenium spots to infer single cell proportions from those tissues. And that's what we are also doing now using Spotlight developed by Mark in Holger's group. And we are combining that also with spatial TCR profiling. And this is done on the same tissue section. So we can actually convert the 3' Visium libraries into 5' TCR libraries, and then, let's say, call the clonotypes per barcode and UMI, and allocate them directly on their spatial coordinates, see where we find expansion of particular clones. And now I will really move from these capture-based spatial tissue transcriptomics approaches to the imaging-based. And so I will be mainly showing data on cosmics. In the previous talk, we've heard about Zenium, and this is a very similar imaging-based technique, let's say. I want to mention here that, so I will show data that has been acquired during the last 3 months. So, but still, in general, imaging-based spatial transcriptomics approaches are in its infancy, which means that there are not yet standardized quality control metrics or computational pipelines that allow us to really, be able to trust on those tools to see what we get from tissue. So what we decided is to actually use tissue that has been really well characterized in Holger's lab, and that's the tonsil. He has talked about that in his talk. And we needed such well-known data set which is also coming from a tissue that's very well structured, very well defined, to actually see how far we could move on with the cosmics instrument. And all the data I will show you has been actually generated by Patri and Sarah, who have golden hands, and golden hands are really a must when you work with archive human material, in order to preserve not only the tissue structure, but also to get good quality RNA measurements out of it. And the data has been analyzed and visualized by Helena, who is a really talented computational biologist who has just also joined the lab. What we first wanted to do is to actually try to see if we could capture like the major populations that we were expecting to find in the tonsil, right? And to do that, we use the in situ type algorithm, which is an algorithm developed by Nanostring. So what we did with that, first of all, it's probably worth mentioning that this algorithm not only relies on the count matrix, but also relies on the background signal, and it takes into account the immunofluorescence data. So from these same tissue samples, right, where we actually are capturing 1,000 different transcripts, we are also staining with immunofluorescence the same tissue sections, which gives us also information at the protein level, information that is also used to then segment single cells, right? So taking into account all these types of information and then also cell size and cell area, the algorithm sort of clusters the data, and you can use it as a fully unsupervised version, or as we did using the human tonsil cell atlas as a reference. And we actually could find all or the majority of the cell populations that we were expecting, and that in a very broad level of annotation, really. And when we plot those populations back into the space, it gives us an astonishing representation of a human tonsil with all these defined, really well-defined structures. And also let me point your attention to the amount of data, right, that we get from every single tissue section. And I will come back to that at the end of my talk. But then apart from looking at very beautiful plots, we can also look at UMAP plots, which we're probably all, most used to look at. And we actually have quite distinct or unique transcriptional signatures from all of these distinct cell populations already defined. And we can also get cell numbers, so cell proportions from each of these populations in every field of view. And if, for example, you look now at this graph and you see that there are a couple of areas with several fields of view in which we have an enrichment with a purple population, let's say. These purple populations are different types of epithelial cells, and if you then look at the global coordinates at the tissue, you see how these purple populations actually very nicely represent the epithelial layer. And if we zoom in into an area of the epithelial layer of this tonsil, we can then better distinguish the exact positioning of any of these single cells that conform these distinct tissue compartments, epithelial cells and immune cells, differentially located in this axis from the basal to the apical layer of the tonsil. We can also look at single transcripts and how the expression also changes across this basal to apical area, which also depicts, of course, the distinct cell populations that we have. And we can also perform receptor ligand analysis on these samples that we actually need to better understand how cells communicate in between them, right? And for that, we are actually using the COMET algorithm, which is an optimal transport-based approach that models competition in between signals, but also takes into account physical cell distances, which is actually pretty important since we have the spatial locations of each of those cells. And you can just, it's not that we need to go into detail on that, but it's just to depict that, so yellow is the ligand of each of those ligand receptor pairs, blue is the contribution of the receptor, and you can see that the different epithelial cell populations actually communicate using different of these receptor ligand pairs, and this actually informs us already about the function and probably the positioning. So if we look at another area of this same tonsil sample where we have 4 very nice germinal centers where we also detect the populations that we are actually expecting to detect in there, we can also look at those tissues, at those areas in a different, let's say, approach, being more, let's say, agnostic on cell types, but really look at gene expression gradients. And we can do that, for example, on a particular axis. If we look now at the gene expression pattern from the marginal zone, entering to the light zone and then until the end of the dark zone, sorry, then we can see how some of these genes are actually upregulated in a gradient-like fashion in one or the other direction. And this will probably become more important when we actually look at tumor settings. For example, where we can see how genes change in a gradient-like fashion from the tumor core to the invasive front, for example. And again, we can look at how cells communicate within these areas, and that's already another proof of principle of how we can see that follicular dendritic cells are actually contributing to the expression of the ligand for PD-1, CD274, while the T follicular helper cells within the germinal centers are actually expressing the receptor. So these 2 cell types are communicating within those areas. And now we are getting to the point in which we are really interested in using these techniques, right, to really understand or try to better understand the mechanisms of cancer development, progression, and metastasis. And that's why we are applying the 1000-plex panel of the CosMx into these tissues that are really challenging to work with, because as you can see, this is lymph node samples, so one would expect actually to have this very nice structured tissue, but of course, since they are coming from CLL patients, the tissue structure has been completely transformed. This is way more homogeneous, also because we have all these neoplastic cells, but still we can capture transcriptional heterogeneity, and this transcriptional heterogeneity is actually linked to the space. And We are also having a look at colorectal cancer samples, both from the primary and the metastatic site, and again, defining cell populations within the tissues, both in the tumor compartment and the tumor microenvironment compartment, and see how heterogeneous those are both in the primary and the metastatic place, and how these different cell types communicate. And all these analyses will help probably or hopefully lead us to a better understanding of how this actually spatially resolved data can be used to inform therapeutic strategies or disease outcomes. And now I'm coming to the last slide of my talk, which is actually which challenges are we facing. And so I pointed your attention already to the amount of data that we are actually generating, and it is just the beginning, right? It's only for 3 months that we are running this. And we have to think about how we will store, how can we process, and how can we share these data not only with collaborators, but with the global research community, right? And we have been putting also a lot of effort on establishing quality control metrics and standardized computational pipelines for these types of data, because they are lacking still. And we've heard from Holger how important that is in order to be able to compare the data across labs, different organs, or even technologies. And just to finish, we are embracing actually the need that we face to establish very close collaborations across the different researchers involved in these analyses, because it's not only that we are dealing with big amount of data, but it's also that we are adding an extra level of information, which is the spatial level, and this really adds on complexity. We have to find ways to better understand, visualize, and interpret this data. And last thing that I wanted to mention is that we actually need more data sets and more diverse data sets to actually learn, test, and be able to use and validate machine learning methods that will help us to agnostically learn from these types of data, but also to be able to stratify patients based on biopsies that actually are normally taken into normal clinical routines, and to help us test several hypotheses. And just to finish, to again emphasize the fact that the spatial multiomics techniques will help us, combined with AI, to better understand the mechanisms behind health and disease. And with that, I just want to acknowledge my former mentor and my former colleagues, and of course, Holger and Helena and all the people in the group, which is really fun to work with, really challenging and interesting environment. Thank you for your attention.
Thank you, Anna. Can you stay here?
Yes.
Questions? Yes, please, Sarita.
Thank you, very nice data. Do you have this developed for organoids also?
We have not tested, but it is possible.
And is it, so do you need, is it fresh organoids what you would need, or can it be
I mean, we can, theoretically, we can use both fresh frozen organoids or then formalin-fixed, paraffin-embedded organoids. We would slice them as if they would be normal, let's say, tissue samples, and we would proceed with them as we do with
Thank
human
you.
material. You're welcome.
Hi, very cool. I really like the gradient of transcription that you were showing. And then at the end, you were mentioning that you need different data sets to learn. Have you thought about using development, so basically embryonic models where you definitely have very clearly, you have the ground truth of, you know, nodal distribution, transcriptional profiles, Wnt signaling, all of these. You know what it's supposed to be like, and to use this as like a, like in the optimization steps.
That's a very good comment, and that's a very good idea, and we'll probably be looking at that.
We'd love to do this together.
Clients, very good. Yes.
And how complex and expensive is to have a custom panel? I imagine the tonsils. I don't want to know, right? But the tonsils is a custom panel based on the data from
No,
products?
so that's a very good point, actually. So what we are using right now is the pan-human 1000-plex panel that Nanostring, let's say, serves. This is not customized at all, but it's still giving us quite good, let's say, biological breadth in the different tissue samples that we've tested. So, and there's a lot more coming in the very near future also. So we are going to the 6K plex panel and even beyond.
Okay.
And that's
Thank you.
coming in the next months.
Hello, everybody. So I'm Bernard Coy. I work at Diagnostics Lonewood. So we are distributors for Nanostring. So as you can guess, this is a sponsored talk, but don't run yet. Okay, so at Lonewood, we've got plenty of technologies that we are distributors, but in my team, we are specialized in Nanostring, Singleton, Twister, and SomaLogic, which are technologies that are directed to research. Why research if we are a diagnostics company? Well, because we need to know what's going to be the next thing that goes into the clinic. So today, I'm going to give you a talk briefly describing these two nice instruments that we've got from Nanostring, which are a case for spatial biology. So today I will focus on the Genomics and the Cosmics platforms. They are both solutions thinking in automation, in mind. So both platforms are really easy to use. So they are also multi-omics, so you can look both at RNA and protein. And they also come with built-in informatics suites, so it's easy to handle these terabytes and terabytes of data that we are going to manage. Okay, so Genomics. Genomics is the whole transcriptome high-throughput spatial profiler. So it gives you, it's an instrument that allows you to detect out to 20,000 genes, working with a variable throughput from 4 to 12 slides per week. And it allows you to work with any kind of sample. It works with ferrous frozen, with TMAs, and with FFPE. Whenever it's coupled with automation, you can do experiment with only 30 minutes of hands-on time. So it's pretty straightforward. And what kind of information you can get? So since you get whole transcriptome, you can get information regarding cell type abundance, pathways expression, so which pathway it's up or down regulated in your samples, and as well as the possibility of doing multi-omics. So with this instrument, it's possible to do RNA or protein or both at the same time. So how does it work? By using standard protocols. First of all, imaging regions. Those are nothing but fluorescently labeled antibodies. Those will allow you to see your tissue and to select your cells of interest. Then profiling agents. Here we are talking about in situ hybridization proofs or antibodies coupled with these reporter proofs which upon the shining of ultraviolet light, specifically only to our cells of interest, those come into the solution and we can pick them up, drop them in a multi-wall plate and do a quantification. Okay, so what kind of analysis we can do? Beyond whole transcriptome, with genomics, we can do RNA assays, such as impulse pathways or cancer transcriptomes, but as well we can work with protein. So for protein, we have more than 500 proteins that we can detect. It's possible to work with modular panels, so we can pick a series of antibodies that have been validated together so that you can go to the results faster. It's also possible to use custom assays, especially for protein, in case you want to detect your favorite or your protein of interest in these samples. Apart from this, I'm happy to introduce the new product that Nanostring is bringing to your research, is that we've got the Immuno-Oncology Proteome Atlas, IPA for short, that it's covering virtually every immuno-oncology protein target in the catalog of ABCAM. So this is a set of antibodies that go all mixed together in the same tube, in which you can prove your samples of interest at once. Those antibodies, they cover all sorts of targets across cancer, metabolism, infectious diseases, organ transplant, etc. And it's possible even to increase this catalog with more than 40 antibodies of your own. So with the genomics, you can take a look at group of cells and compare one group of cells, one group and another group of cells. You can do so by selecting for fluorescence, by using these fluorescent markers, or by geometry. So you can take a look at a specific group of cells and just select them by, imagine that you are using ImageJ or Paint for doing a selection for these cells, and then you press a button and you collect those cells and get this highly enriched sample in which you can do your analysis. This is, for the genomics, we are talking about multicellular level, but for the cosmics, as we have already seen in this session, we are able to locate specific transcripts on each one of the specific cells of our tissue. So we are able to analyze these millions, hundreds of thousands of cells, and by doing an in situ hybridization technique, locate those transcripts on top of them. We can do that for RNA, but in the thousands of duplex. By the beginning of next year, we will be able to do 6,000 plex, and more coming soon. And for proteins, we can do up to, well, we've got a panel for 64 proteins, and this will be increasing soon as well. And how does it work? Well, I took the time so we can go a little bit deeper into the chemistry of this technology. So Cosmics uses in situ hybridization chemistry. RNA or protein targets in the individual cells are identified through the hybridization of specific proofs or antibodies which have a special readout domain. Okay, so we are going to do this the readout domain. We're going to use this readout domain to be able to decode for all the specific transcripts or proteins that we want to detect in our samples. Cosmics is special in that sense because we are able to use, we only need up to 5 proofs per gene. Those proofs are really small, so we can spanning only from 30 to 50 nucleotides. So we can work very nicely with partly degraded RNA, which makes it very suitable for working with FFPE samples. So with the genomics, with the cosmics, sorry. You get different layers of information. You can get the protein expression. You can get the RNA expression. You also get the cell segmentation because it's crucial to know the boundaries of each of the cells to determine whether one transcript is part of one cell or the neighboring cell. As well, with this information, altogether, you can do cell typing, receptor interactor analysis, etc. So let's take a look what happens if we take 2 serial cuts of tissue. In this case, today we are talking about lymph node biology. So here we can see the typical image that comes out of the Cosmics. You can see these little squares are the fields of view that you are selecting, and you are analyzing all the cells within each of these fields of view. On the other hand, on Genomics, I don't know if you can see it from here, but there are these little structures in which there is a predominant color, which means that there is a predominant cell type, and you can do this geometric segmentation and pick specifically all the cells that are within one geometric area, or that have increased fluorescence for one or another marker. So by integrating both technologies, you get different information. For example, we can get the who did it and what happened. Here I'm showing you by Cosmics data that, for example, CD86 from beta cells colocalize with CD28 T follicular helper cells in the germinal center. So in this kind of diagrams, you can see that one cell that is expressing a ligand, it's close together to one cell that is expressing its receptor. But you can dig deeper on this information by using Genomics. With Genomics, you can tell the differences in the different, well, here we're talking about germinal centers, so we've got dark, intermediate, and light zones, and we can see the different expression in between of these areas. So we have the single cell information, which can be used to check what are the cells doing altogether, as well as the whole transcriptome information, which helps us to discover these genes that are up or down regulated. For example, here we can see that there is an interleukin 18 in cells close together with which are expressing CD48. And what else are we seeing? Well, we can see that, for example, in these germinal centers, interleukin 18 expression comes from the myeloid, from cells in the myeloid lineage. Those, well, this is an example on how you can use both platforms. Another example could be, for example, for doing a spatial analysis for one sample. You can use the Cosmics to do quantitative cell profiling, whilst you are using the Genomics for doing, I don't know, for example, for predicting immune signature profiling. To sum up, Nanostring has provided Atomics, which is the platform which will help you not only to analyze the data, but also to share it among your collaborators, so that we can share these efforts and push forward these discoveries. So thank you very much. This is all. Here we've got the contact information, and if you're interested in these technologies, please come and say hello, and we'll be more than happy to help. So thank you very much.
Thank you very much, Bernard. Are there any questions? Yeah, you see here.
Just one question. Which is the minimum number of cells that you have to select in the Genomics in order to be able to actually get enough material?
Yeah, it depends on whether you are doing RNA or protein analysis. Mm-hmm. For protein, it's about 20. For RNA, 100, 200. It also depends on how transcriptionally active are these cells.
And is there also, let's say, limits on the area that you want to profile in terms of, big or small?
Yeah. For small, there is no limit, right, because in fact you can pick cells that are scattered along your tissue. Imagine you've got a CD68 for macrophage, mm-hmm, and you can pull them together as long as there is enough of them. If you are falling below this limit, maybe you will get a less signal for these low expressors. And regarding size, there is a maximum size, which I guess, if I recall correctly, it's like 600 micrometers square,
Thank you.
more or less.
Yeah.
Welcome.
Okay. Thanks so much.
Thank you.
Thank you. Okay, so my name is Yosune. I'm a medical oncologist and I work in BHIO in the colon cancer unit. These are my disclosures, but my main disclosure is that I'm a physician, so I'm gonna talk from the clinical point of view during this talk. So as I said, I work in the colon cancer unit, and well, colorectal cancer is the third most frequent tumor worldwide, but it has to be said that it's the most frequent here in Catalonia. And when it comes to mortality, it ranks second. The mortality, thanks to the improvement in cancer management, has declined in developed countries, but what we see is that the incidence is increasing, and this may be due to different causes. The first one is the screening and the early detection programs that makes that the incidence goes up, and the second one is that we see it is increasing, and this is due to the westernized and the dietary patterns and the lifestyle that we have nowadays. Here, this is a really brief overview of the clinical management of colorectal cancer. It has to be said that the backbone of the management today is chemotherapy, more specifically the fluoropyrimidines. And it's been only in the recent years when we have been able to develop targeted therapy for patients with colorectal cancer, with drugs for patients with RAS, BRAF wild-type tumors, for patients with HER2 alterations, for patients with BRAF mutations, and even it's still under development for patients with KRAS G12C mutations. But when it comes to immunotherapy, and in contrast with other tumors such as melanoma or lung cancer, we are not as lucky because we have been only able to develop immunotherapy for patients that present MSI tumors. So we can divide our patients in patients that present tumors that are MSS and patients that present tumors that are MSI. This means that for patients with MSI tumors, they present a deficiency in the machinery, in the mismatch repair machinery, and these tumors are going to present what we call a high TMB. That means that they have a high tumoral burden, they have a lot of mutations, and this means that those tumors have a high infiltrated immune cells, and that they are what we call hot tumors. Meanwhile, patients that present MSS tumors, they don't have any deficiency in the mismatch repair machinery, and those tumors are going to have a low tumor mutational burden. They are not going to have so many mutations, and these tumors are cold, and the immune infiltrate that we can find in the microenvironment lacks active T cells, while they have a high number of Tregs. So this classification can translate into a different response to immunotherapy. You can see here that for patients with MSI tumors or that have the mismatch repair deficiency, they are going to respond to immunotherapy, while the other patients are not going to respond. And it would be nice if this meant that these are half of our patients have MSS tumors and half of our patients have MSI tumors, but the truth is that only 5% of the patients that have a colorectal cancer have MSI tumors, while for the other 95% of our patients, immunotherapy is not useful at all, at least today. Here you can see in this table the response that clinical trials trying to prove the efficacy of immunotherapy had, and they are mostly 0%. Only in some cases we saw some responses, but that was not enough. So it is time to move forward and think how we can change that microenvironment of these tumors to make them responders to immunotherapy. So how can we move forward? I have, or the clinical community has 3 ideas. The first one is designing novel clinical trials with a different design. The second idea would be trying other compounds or other treatment combinations. And the last part would be other novel settings and thinking outside of the box. So the first one, novel clinical trial designs. It was seen in this clinical trial. This is a clinical trial showing the efficacy of 2 drugs. One is botensilimab, which is a CTLA-4 inhibitor, and the other one is balstilimab, a PD-1 inhibitor. These 2 drugs are not the same as other CTLA-4 inhibitors or PD-1 inhibitors. They are supposed to be more efficient. And what this clinical trial showed is that some of the patients responded, around 24 of the patients responded, but when we focus on the patients that didn't have liver metastasis, liver involvement, we see that this 24% of response is higher, and it comes to be 42% of the patients responded. So now we have designed a clinical trial that is proving or trying to prove the efficacy of these 2 drugs, but only in patients that don't have liver metastasis. It has to be said that liver metastasis is the main organ, liver is the main organ where patients with colorectal cancer has metastasis. So it's very rare to find these kind of patients, but the clinical trial is ongoing, and in the clinics what we see is that all of them are responding. And why is that? Well, this is because we have seen that liver metastasis may be mediating immune resistance, and that is the reason why they do not respond. And this is something that we are investigating. We are investigating in collaboration with Holger's lab here. And what it could be, it has been postulated, is that there might be some interactions between the tumor cells, the liver stroma, and the immune cells that are in the tumor, and that the hepatic or the liver macrophages may be inducing CD8 positive T cell loss, so they would be inactive. Another clinical trial that can be designed with a novel perspective would be the use of single cell in the clinical trial. And in this way, we have designed the Serpentine study. This is a clinical trial that is testing the use of two drugs, durvalumab, which is an anti-PD-1, and tremelimumab, which is an anti-CTLA-4. And what we do is that right before starting the treatment, when they have started the treatment at maximum response, and when they progress, we perform a biopsy to these patients, and we do single cell analysis to see the immune cell atlas of these tumors, and to see how are those cells and why some of them are responding, why some of them are not. What I see in the clinics is that the weak point of this trial, it has to be said, is that it is not easy to biopsy a patient, and it has some risk. And the easiest organ to biopsy is the liver, and we know that is a marker of immunoresistance. So it would be very nice to have patients with involvement in any other organ that could be biopsied, but well, we have to wait to see the results. And the last novel clinical trial design that I would like to show you is the use of immunotherapy in combination with temozolomide. Temozolomide is a drug that is not, its use is not very extended in colorectal cancer, and we only have some evidence, low, medium evidence from phase 2 trials that have shown that it could be used in patients that show a silencing of the MGMT promoter, and in these patients, temozolomide could be used. The problem with temozolomide is that at some point the patient is going to progress, and this is due to an acquired resistance to temozolomide. That what we have seen is that this is mediated by mutations in the MMR machinery that I mentioned at the beginning. So what this means is that these tumors at some point belong hypermutated and they behave as if they were MSI, which is something that could be seen as something bad, but if we take cautiously, that could be also something good. Why? Because we know that these patients might respond to immunotherapy. I mean, patients with MSI tumors or patients with a phenotype that simulates MSI tumor. And this is some data that was presented by the group of Luis Diaz from the memorial in ASCO last year, where they presented, as I told you, that a low TMB, it is a biomarker of immune resistance. But what they see is that it's not only the low tumor TMB, but it is also what they call the alien peptide score. This means that it is not only important the number of mutations, but how those mutations are. And this means that the more different the mutations or the peptides or the proteins are from the normal and regular ones, the more chances of the immunotherapy being effective. And based on all these, they have designed 2 clinical trials that are called the Maya trial and the Aretusa trial with similar rationale, because what they do is that they give temozolomide to patients that have MGMT methylation. And in one of them, once they have progressed, and in other ones, they have a high TMB, they give immunotherapy, and what they see is that patients that have a high TMB after having temozolomide, then these patients are going to respond to the treatment. Our next setting that I want to talk to you about is the novel compounds and treatment combinations. As I told you, the use of immunocheckpoint inhibitors are not useful in these tumors or in these patients because of the biology and because of the microenvironment. So this is a bunch of clinical strategies that have been designed for these tumors. I want to focus just in some of them. The first one would be the combination of the immunotherapy with targeted agents, because what we have seen is that patients that present alterations of the MAP kinase pathway, those tumors present a lower immune infiltrations. If we get to inhibit that MAP kinase pathway, we get to make those tumors hot, and then the immunotherapy could be useful. Here you can see in the left graphic how these tumors, once the patient has started the treatment, the immune infiltrate that these tumors saw is higher, and in the right you can see how the tumors responded, and that always, or in most cases, or for those lucky patients, translates into differences in survival. The other strategy I want to talk about is the biospecific antibodies. This is novel strategy that is based on the use of a biospecific antibody that is going to bind to 2 different antigens. One is normally going to be present in the tumor cell, and the other one is going to be present in some point or in somewhere in the immune cells. In this case, the cibisatamab is a biospecific antibody that binds to the CEA, that there is a protein that most colorectal tumors express, and it is going to bind also to the CD3 that is in the lymphocyte. And here you can see in the right graphic CT scan images from a patient that was treated with this drug. It's only a drug, even if it's binds to different points, but it's only a drug. And this is a patient with an MSS tumors, and here you can see how those lung lesions that were present at baseline, after 16 weeks of treatment, they mostly disappear. And this is something when you are in the clinics and you have treated many patients and you have seen that many of these drugs have failed. This is not like you got to cure cancer, but it's like you feel that you have made one step forward, and it's very, you get very happy seeing this. And the last novel compound on treatment combination that I want to show you, and well, I'm going to be very briefly about this because these are some drugs that are in very early clinical development, but these drugs are going to be directed to different points of the immune microenvironment, again with the same idea of getting to make those tumors hot. And as I told you, the last part of my talk is going to be about novel settings. And here I want to focus on the microbiome because it is something that we in the VHIO, in the colorectal cancer unit, we have a very special interest. The microbiome is something that is, sometimes we think that is something very new and fancy in oncology, but it has to be said that is a really old story because we know many viruses or bacteria that may be causing, or that are actually causing cancer, like HPV and cervix cancer, HIV and Kaposi sarcoma, or Helicobacter pylori in gastric cancer. But when it comes to colorectal cancer, the story is more novel because we have only been able to identify 3 oncomicrobes that could be mediating or could be involved in the colorectal cancer pathogenesis, and are the Fusobacterium nucleatum, the PKS positive E. coli, and the enterotoxigenic Bacteroides fragilis. And we have always the same doubt if this is something causative or associative. We don't know if what is first, the chicken or the egg, and that is something that we are trying to elucidate. And we also want to see its role in immunotherapy because it has already been reported in other tumors like lung cancer and melanoma. Those tumors also always go ahead of us in this setting. But there is a rationale to think that there could be some interactions between the microbiota and therapy. And we know that Fusobacterium nucleatum at least has a role in persistence and recurrence of rectal cancer. This is something that we published in Annals of Oncology 3 years ago when we saw that those patients that were still positive for Fusobacterium nucleatum after being treated with chemo and radiotherapy, those patients presented higher chances of presenting progression disease or recurrence. Here you can see in the ATEZO-TRIBE that is a trial that tested the combination of chemotherapy and immunotherapy, that they detected some bacteria, more specifically the Intestinobacter E. Collinsella, that their absence or presence was related to better outcomes. And we have more interest in the Fusobacterium. This is an investigation or a trial that is currently ongoing, where we are seeing the presence of Fusobacterium in patients whose tumors were detected in the screening cancer program, and we are seeing which patients present the Fusobacterium in the biopsy or in the tumor, and how they behave, if they progress, if they don't progress, if they respond to chemotherapy or immunotherapy. These are some partial results where we see that there are differences, at least when talking about the states and the location of the tumor in the colon. But the real question is if we can change the microbiota to make it work for us. I mean, to make the treatment more better for our patients. And here you can see some strategies that we could use, as the use of antibiotics or prebiotics or probiotics, the use of vaccines, the use of some bacteria that could work to kill other bacteria that could be mediating immune resistance or chemoresistance, and finally, the fecal microbiota transfection. Here you can see a work that was performed in our lab some years ago, where we give to some mice, to murine models, metronidazole to treat Fusobacterium, and what we see is that for those mice that were Fusobacterium positive, when they were treated with metronidazole, the tumor volume, the rate of increase in the tumor volume was decreased. And finally, the fecal microbiota transfer. This is a trial that was designed for patients with melanoma again, and what they saw is that this is a strategy that could be used. It has to be said that it has to be done cautiously, and it cannot be done in every center, but it has to be done with very high measures of security, because it is something that can really can be very dangerous for the patients. But an idea that I want to give you and to transmit to you is that the study of the microbiome is very complex, because you need a lot of samples, and they are not only tumor samples, but you need also fecal, oral samples, and you always have to be able to study the exposomes. That means all the factors to which the patient has been exposed throughout all his life, or their lives, because this could be affecting or changing somehow the microbiome, and this is something that is not easy, and it means the use of very, very long questionnaires for the patients that they are very nice and they complete, but it is complex. And finally, just an idea about the Optimistic project. This is a project that is funded by CR UK, and that we are doing in collaboration with Harvard University and Dana-Farber Institute and many other centers, but in which we are studying how the microbiome changes in patients that receive chemotherapy, immunotherapy, and for patients that present what we call early onset disease. That means that the diagnosis has been made below the age of 50, because we are seeing that it is the incidence of colorectal cancer is increasing in the young population. And what we do is that we collect samples and questionnaires about lifestyle in all the points of the disease to see if there are changes, if we can correlate that with response and with toxicity. These are some preliminary results where we can, we have to analyze better, because this is like a raw analysis where we can see that we can detect the gut microbiome, and that there are some changes that may be affecting the state, the location, where the patient is from. I mean, patients from Dana-Farber and patients from Barcelona and BHIO have different microbiome, but we have to go deeper in the analysis. Now we have 1,500 samples, or different patients, better. I mean, we have many more samples, but we have to go deeper in the analysis. And these are my conclusions. What I think is that, well, the tumor for sure has dynamic changes in the tumoral cells in themselves and in the microenvironment, and that sometimes means resistance to treatment, but that also means new chances to change it and use it for the sake of our patients. The idea is also that it is really important to collect samples, but it is also true that that is also sometimes very difficult. Our patients do their best, for sure, but we sometimes ask too much, it has to be said. And thank you very much.
Thank you very much, Josina. Don't stay here. They're going to ask you questions.
Okay.
So, do we have questions? Yes, please.
Yosune? Okay. Thank you so much for your presentation. It was super interesting. I mean, I touched mostly the last part of your presentation because we work microbiome every day. And my question was related to what kind of analysis did you perform to get your results? I mean,
For the microbiome?
Yeah. Did you focus your attention on 16S metagenomics barcoding, or also you are thinking to use shotgun to highlight maybe the presence of some genes related with the colorectal cancer? Thank you.
Well, in fact, we are doing both. These results that I saw are from the shotgun, but we are doing 16S and shotgun.
Thank you so much.
Hi, this might be a very naive question, but so it makes a lot of sense to me that you look at the microbiome for colorectal cancer. Did I understand correctly that you do so that there are being done fecal transplants for melanoma as well, and that this could have effects on the outcome? And then if I understood this correctly, then how does that work, right? Is there a systemic effect of the fecal transplant, and could this be detected in plasma or in the blood of patients?
Okay, so from the fecal transfer, that is something that we are not doing in our center. As I said, the fecal transfer is very complex, and it has to be done in ICU units because, I mean, some patients, a fecal transfer has led in some patients to sepsis and death, and it has to be done very cautiously. In this clinical trial that was performed in other center, what they did is that they transfer fecal material from patients that have responded previously to patients that had not responded, and
but in melanoma.
So melanoma, yeah, with melanoma.
So then, I'm just interested about in the intertissue communication axis there. Right. So, and because there must be like from the fecal material, which is presumably in the gut, there must be like communication to the skin, and then this must be a systemic effect that possibly could be measured.
But that makes sense because everything that we do affects our microbiome. It's not only what we eat; it's like how we were born, what if we do a sport or we don't. Then there is some communications between there is some communication between the microbiome that it is in our gut and the rest of our body.
Thank you.
And that was made in the response. Yeah.
Okay, thanks.
Hello, thank you very much for inviting me to present at this conference. I'm really sorry I cannot be there. Unfortunately, a double line on COVID testing this morning means that it's safer for everybody if I take this from my house. Anyway, I'm here to talk briefly about what Illumina does and what the recent innovation we're putting together and share them some with you. So my name is Pietro Gatti. I've been in Illumina for a fair bit of time, and I spent most of it in R&D when I was leading the development of our technology and our platforms. Now I'm in a strategic role within a commercial organization, which basically allows me to come here, well, virtually in this case, and discuss what's to come with you and her about your feedback and your desires for the future. Today, I really want to talk about innovation at Illumina, and because I have a very limited time, I'm only going to talk about two aspects in particular. So what the XLEAP SBS chemistry is, and then later on about complete long reads. But before I do that, I really want to spend a couple of time talking about or reminding everybody what we do at Illumina in terms of innovation. This is a slide that I'm sure most people in the audience are familiar with. Illumina over the years has created this incredible portfolio of instruments that really cover all the different ranges, throughput, application, arrays, and sequencing. And this is just the sequencing part. Of course, we have all the library prep, all the data analysis, and everything to go with it. And this doesn't really just happen. There's so much effort and so much innovation that goes in it that I really wanted to highlight in a little bit. We said Illumina innovation is in our DNA, and this is quite true actually. We spend a lot of time thinking about what is that is needed in the market and by our customers. And you can see here some snapshot from application in terms of what we've done with the Primate AI 3D publication early on to really create the information needed for people to make the most of NGS. You can see down there on the left, one of the new library prep kit dedicated to oncology, which provide unparalleled access to a lot of information in samples. And then there is all the effort in terms of integrated solution that go from the planning, the sequencing, the library prep and the analysis and the interpretation of the analysis that we're providing. And here on the right, you can see another case in which is a response to a very practical need, which in this case was the rapid turnaround time for the respiratory virus panel detection with the mini-sig rapid configuration. So this is really just to give a snapshot of everything we're doing, and I really want to focus on a couple of these. But before I do that, I want to actually put in perspective what this rate of change means. So what you can see on this slide is the first platform, high throughput platform that the Solexa, just before being bought by Illumina, put on the market in 2007. And then on the right side, the NovaSeq X platform, which I'm going to talk a little bit more in a short while. What I want to show you, if you look at the cost per G, we go from $3,000 in 2007 to $2 per G currently, which is a 1,500x reduction. Conversely, if you look at the throughput, we go from 0.3 gigabases per day in 2007 to 8 terabases per day now, which is a 26,000x increase. Now, to put that in perspective, if sequencing was a plane and we were to fly between New York and Paris, we would go from doing it for £2,000 and 6 hours in 2007 to just a few dollars and a few seconds currently. And this is really to put in perspective all the innovation, all the effort, all the development that went into the Illumina product and what that means for customer, for all of you and all of us. Now, moving on, I want to spend a little bit of time on XLEAP SBS chemistry, and in particular, describe a little bit why we think it's such a brilliant idea to put it on our sequencers. So, in a nutshell, XLEAP SBS has been already discussed and presented a year ago. It's really the next iteration of our sequencing chemistry. It consists of new compounds, new reagent, a new integration of the reagents, a really a new reinvention of what the SBS chemistry is. With the idea of delivering what you can see there on the right side, faster cycle time, improved performance, and more sustainably. In particular, on the NovaSeq X, as you know, there is this lyophilized and stable reagent that allow a whole new way of handling our reagents as well. So, what does it mean for our sequencers? NextSeq 1000 and 2000 we'll see XLEAP chemistry coming into all the flow cells and all the configuration next year. So this is already a platform that has been really transformational for benchtop sequencing. It's flexible, it's scalable, is a very streamlined workflow, has the greatest quality in Illumina we can have, and now it comes with XLEAP. So what should we expect from this? I want to show you a couple of data points that really highlight why XLEAP SBS is the next chemistry for Illumina. What you can see here on the left is a like-for-like comparison of the same flow cell, the same configuration, a 2x150 cycle run, done with the two chemistries. As you can see on the left, the throughput is not going to change, and that's perfectly to be expected because the chemistry is not going to change the flow cell by itself. But what you can see in the middle and the right side of this plot is the impact on error rate, which, as you can see, drops between 30 to 40% moving to the new chemistry, and also runtime. XLEAP SBS is yielding a 20% runtime reduction on the same configuration, which is very appealing, and I think is going to make a big difference for our customers. I also wanted to show you some of the early data we've seen for 600 cycle kits, where XLEAP SBS chemistry is delivering a substantial decrease in error rate, meaning that this chemistry is performing incredibly well, and particularly so at longer cycles. There's a lot of discussion about quality scores, and so on, and this is a very long conversation, but I want to show you this snapshot. When we look at individual based Q-score bins for our runs, which, as you know, usually are reported in terms of ranges of Q-scores rather than individual ones, we can see that with XLEAP SBS on a NextSeq 1000 and 2000 platform, the distribution shows that most of the base scores are above Q40, so really yielding the highest quality we can expect from the system. Moving on, the NovaSeq X series, again, this has been in the market for a year now. It came out straight away with the XLEAP SBS chemistry. As you can see on this slide, there's a number of incredible features on this platform, and what it can deliver in terms of throughput and cost reduction and sustainability are really unparalleled. And as you can see from there at the bottom, SBS is clearly one of the many innovations that went on this platform. And this is really a point I wanted to make. So when we think about the chemistry and we co-develop it with the system, then we can think about delivering even more benefits than when we previously could. And I think this is particularly well demonstrated by the sustainability aspect. So actually SBS, together with lyophilization, enabled a new way to think about ambient shipping of our reagents. And the picture you can see on the right side of this slide, I think is very compelling. This is comparing the sheer volume of all the reagents needed for a NovaSeq 6000 system run compared to what the NovaSeq X series will deliver. Besides, the ease of handling all of these, the space requirement, and so on, I think the impact on sustainability is something to be really happy about the integration of the chemistry, the innovation, the lyophilization, and the architecture of the instrument. And here again, I did want to talk about the quality aspect of it. In this case, the plot is slightly different from the one you showed before. So in blue, you can see how the customer-facing bins have been reported now on NovaSeq X. So in the new software release, we've readjusted the distribution of the bins, and now the bins reported are going to be called around the Q40 score. But what I wanted to insist on a little bit more is that even though in reporting Q scores, by necessity, we need to use ranges because otherwise we'll be just too computationally intensive. If you look at the underlying distribution of Q scores, again, you can see that the vast majority of base scores are well above the Q40 mark, which yet again highlights the quality of all Illumina sequencers and even more what XLEAP SBS can deliver. Now, I think I'm running a little bit of time, out of time, so I'll jump into the complete long reads part of the products. Again, here we're moving from core sequencing into application and library prep. This kit has been already announced a year ago in the sense of the whole genome solution. And here is just a slide to remind us that Dragon, together with short leads, is addressing the vast majority of all the problems in the genome. And the improvement we can see by combining the chemistry and the analysis it keeps improving and is yielding the best genomes we can think of. However, Illumina has also introduced this complete long reads solution to expand the capabilities and address a small part of the genome that remain challenging for shorter reads. And you can see here a snapshot of which ones they are. What I really want to focus on today, though, is the upcoming release of the enrichment version of the Illumina Complete Read Prep. You can see there on the left, I think this has been shown before, I'm sure many of you are familiar with it, but just the concept of what the enrichment panel does. So you can see at the top is the traditional snapshot of what the short reads from Illumina will generate. In the middle, the whole genome version of the complete long reads yield a fantastic improvement in coverage across the whole genome. And at the bottom, what the enrichment version does. And I think this is really the appealing part of this solution because it allows people that know what they need to look for, which part of the genome they're interested, to really zoom in and get the best possible coverage of the region they're interested in. So what is that going to mean in practice? That there is this human comprehensive panel that is going to be launched early next year. As you can see, it covers a specific region and really enhance the mapping of challenging regions. But also, as you can see on the right side of this plot, just behind my image, I'm afraid, a 50% reduction on the scoring in terms of false positive and false negatives. But this is not it. There's a roadmap associated with this product. Now the Human Comprehensive Panel is coming out. There will be a number of pre-designed panels which will allow customer and people running this as a service to really zoom in other areas they're particularly interested in. And furthermore, with the possibility of designing custom panels, I think this will additionally improve the ability to zoom in into region of particular interest. I believe that this is going to be a great product that is really going to deliver enhanced capability for our customers. I'm afraid this was a very quick presentation. I didn't have the time to cover more topics. I really wanted to give a snapshot on what Illumina does in terms of innovation and use XLEAP SBS and complete long reads as an example. Again, I'm sorry for not being able to be there to continue this conversation with you. If you are interested, please reach out to the local sales representative, or you can also reach out to me at this address. Thank you very much, and I'll talk to you next time. Goodbye.
Have done tremendous efforts on integrating Hi-C into the assembly. But I'm going to bring you a different aspect of how we look at the genome in 3 dimensions as part of what we call the Genome Center for Imaging the Genome, the Center for Imaging the Genome, which is a collaboration that we have paid by the NIH. It's a large NIH grant between these 4 labs, and it's coordinated by Tim Wu, who is the person up here. Let me see if I have a pointer. Yeah, by Tim Wu, who is coordinating the CGI, the Center for Genome Imaging. Now, what we want to do here is the objective of the CGI, which is finishing in the next 2-3 years, is to be able to develop technologies that would allow us to look at the genome in 3 dimensions, the entire genome at high resolution and with as many cells as possible at the time. Normally, what you would do in imaging is use FISH, which you have access to few different points of the genome by hybridization, at most 4 or 5 at the same time, but you can do with a lot of cells. However, we want to also target many different targets, and I'm going to talk about oligostorm in the next few slides that allows it to do super resolution imaging of a number of targets, not very large numbers, but a number of targets in the genome. And then in the second part of my talk, I will talk about OligoPhysics, which is a multiplexing system that allows you to see thousands of sites of the genome at the same time, and in a large number of cells, however, with low resolution, okay? So these are the 2 talks. Let me start with oligostorm, which is chromosome walking at super resolution, and we combine here also Hi-C to increase the resolution of the data. This is something that was done by Guy and Irene quite a number of years ago, but we have been improving the methods. You can read the original paper here in 2018, but we have been improving the methods, and you will see at the very end that we are also combining this with OligoPhysics. So how oligostorm works? As you can imagine, we need oligos. These oligos have a homologous part that is targeting the region of interest, so you have to make a design of region of interest. They have a main street and a back street, and this main street is used to attach a secondary oligo that then will blink under the microscope. We do that in this microscope that you will see if you visit the facility at Sinak. It's a Bruker-Boxell machine. So what we are going to show you now is the walk of these about 8 megabases in chromosome 19 in human cells in 9 steps. So this will be the secondary oligo that goes and blinks. So how we do that? The first thing, using the fluidics machine, which is that part here in the machine, we throw all the oligos at once that cover the entire region of interest. But then, step by step, with secondary oligos, we blink the first segment, then we blink the second segment, and then finally we blink the end segment, in this case 9. So in this way, what you can do is by sequentially analyzing each one of these blinkings, you can walk a chromosome, in this case 8 megabases. So you separate the signal in time rather than in space only. We have 9 segments that goes in this particular scenario. We did it for studying compartmentalization of the genome. I'm not going to go into the details of the biology why we selected this, just describing the technologies today. These are the number of oligos in each one of the sites. They cover hopefully as homogeneous as possible the region. These designs are not trivial because some regions are very repetitive. You cannot have, for example, in here, very repetitive regions. You cannot have oligos there. But anyway, this is the distribution we did back then. And this is how it looks like in one cell. What you see here is one of the cells that we image. And what you see is every time you see new blinks, it's one of these segments that you separate over time. And what you have here is the 2 copies of chromosome 19, one of the homologs and the other homolog. And you can see right away that the structures are quite different. And this is something that we know for many years, that the structure of the genome is very dynamic. It changes even between the two homologs. You have very different structures. But then we take these blinkings and we transform them in something that is a density map. And these density maps then are used to increase the resolution of the data so that we can try to trace the chromatin as it goes through these 8 megabases. The way we do this is using TATBIT, developed in our lab, and using high-C data. And we transform contact maps into 3-dimensional models using TATBIT. And then this is segment number 3, the area that is blinked under the microscope. And then we take all these models and we feed them inside. You probably have seen it before. This is the distribution of the fitting of all these models inside, and there were a few of them that fit pretty well into the data of the single images. Remember, high-C is a population-based. It's an average of all the cells that you have in the experiment, while this is a single image of one cell. And then we also use flexible fitting to increase the fitting of the model inside the blob with no increase in the clashes or the distortion of the high-C contact maps that we have used in the modeling. In this way, we can finalize a 10 kb resolution, the path of the chromatin in the 2 copies of the chromosome 19 in that particular cell I have shown you before. With that, now we know which parts of this blob correspond to what chromatin, and we can start looking at promoter and enhancer interactions in these particular cells, the variability between cells, etc. Unfortunately, this is not very high throughput as of today. We're working to make it more high throughput, but we can get tens of cells only, so the statistics are still weak in reality. That's why we also help developing oligophysics that you can go to hundreds and thousands of cells, unfortunately not at high resolution, but at low resolution. So this is the work we did in 2020. You can read more the details in this Nature Methods. Again, it's a collaboration with the people at the Center for Genome Imaging, the work by Hugh, Antonio, and David in this case. So let me recapitulate. We are looking now at this, looking at many sites and many cells, but not high resolution. Here, as you've seen, the previous paper was 2018. This is 2020. The oligos have improved. We are now designing the oligos in a way that you can have not just a main street and back street, but you can do quite a bit of different things in each one of these branching of the oligos or the streets. And they have the same. They have a genome homology part and then things that you will do with the other parts of the oligos. They go into a particular region of interest. You can have many regions in the genome, up to 1,000 regions in the genome. It's a multiplex effort. And then what you do really is the same that you would do under the Illumina microscope, right? Because Illumina at the very end is a microscope as well. You're gonna sequence the oligos that you are putting next to the site of interest. So you are targeting a site of interest, and then you have designed the oligo in a way that you sequence the oligo and you have a specific barcode for the site of interest. If you follow me, in fact, it can, because of the design of the oligos, it can be used by ligation-based, synthesis-based, or hybridization-based. What you're going to be seeing now is SIT, synthesis-based identification of targets. But any of these 3 types of chemistry would work equally well. These are just to show you that we can detect the signal in more than 98% of the times. Okay. Now, what is the advantage of doing this multiplexing? You will figure it out. In normal fish, what you have is the number of targets that you can target is equal to F by N, which is the fluorophores that you have and the rounds that you do. So it doesn't scale exponentially, it scales linearly. With now the barcode sequencing, what you do is you put the N to the square hub here, so the number of fluorophores multiply the number of rounds that you have. So what I'm going to try to figure it out is what these particular 4 colors correspond to a particular site of interest in the genome. That means that you can scale pretty quickly in number of rounds on this sequencing. So in 6 rounds, for example, you can have about 4,096 sites in the genome. In 7 rounds, you get up to 16,000 sites in the genome. Now, is this real? You can actually do it nowadays? Not really, because the signal, when you have so many signal blinking at the same time in such a small space, it's very difficult for us to differentiate who is who. But we have done 1,000, so 4 rounds to 5 rounds, so 1,024, and we can do pretty well that, which means that, for example, the fly genome could be covered entirely at somewhere in between 4 to 5 regions if we want to cover it at half a megabase, okay? So we would see all these half megabase sites in the genome of flies in only 4 to 5 rounds. The human genome needs to be somewhere around 6 to 7 rounds, and we're working for that to happen. So the proof of principle, it does work. Well, it does. Let me show you what we did. We took 36-plex. This is a 36-plex. There are 6 sites in 6 chromosomes across the human genome. Each one of them have different sizes. We use 5,000 oligos per target. There is about 7 to 70 megabases between them. As you can see, we try to cover each one of these 6 chromosomes more or less homogeneously in each one of the arms with these 6 sites. And then we put them under the microscope and we start collecting the data. This is round 1, round 2, round 3, round 4 of sequencing. And then you have the different channels of colors, 4 channels of colors. And you will, and this is the merge of the 4 channels of colors. And you can see that, for example, this point here is purple. Then in the round number 2 is green. Round number 3 is green. And round number 4 is purple. And the only oligos that we designed to be purple, green, green, purple corresponds to this site in the genome. And that is how you differentiate the different sites, by this barcoding of the oligos. Another way of putting it is in here. This is not real. We just put them one on top of each other of the rounds. You see the rounds differently, but this one, for example, is all the 4 rounds yellow. This one is cyan, red, cyan, cyan, and so on and so forth. In each field of view, you have a number of cells, and you can do many field of views and cover hundreds to thousands of cells in this approach. By the way, this is 3D, so we do Z-scans as well, so we have the 3D organization. How well it works? Well, we can identify in this 36-plex, the majority of the sites, we can identify more of 80% of the cells contain the barcode that we expect, because they should all contain. For whatever reason, in this experiment, chromosome 5 gave us problems, so we have to rethink how we actually define the oligos that goes to chromosome 5. And this is one of the tracings that you can do now. For each one of the chromosomes, you can see that we find where they are and the tracing of the 6 points of each one of the chromosomes in this particular cell. Now, this was the 36. We could do also a 46-plex in one single chromosome. That's chromosome X. That would be the equivalent of having thousands of points for the entire genome. And you can see here, this is the location of chromosome 6 in this particular cell. And this is the tracing that we can do in 3D. So we believe that we can do thousands of sites. Well, the maximum we have done so far is 1,024 plex. And it's difficult to differentiate the signal, but it's possible to differentiate the signal for the majority of the cells. So what are the advantages? Is that oligophysics is relatively fast for what it means imaging. In 2 days of image acquisition, you can get 1,000 cells. In our case, we had about 5,000 complete chromosomes. So my group is interested in chromosome architecture, so we can study now statistics on how the chromosome folds. And then we had about, of this 1,000 cells, only 150 cells with complete 6 chromosomes. That's also a difficulty here, to have all the signals in each one of the individual cells. It's also complicated, and here you have several examples of such a thing. The things that we're working currently is not yet ready, but probably this year or beginning next year, is combining oligostorm and oligophysics in one go. This Butara voxel machine can do both, can do wide field and super resolution, which means that you can do a first round of oligostorm and get high resolution data, super resolution data. We don't know who is who in here. We just take all the blinks at once without doing the steps. But then on the same sample, we do oligophysics with 2 rounds. We can differentiate who is who, and then we just color by who is who the data, and we can have a super resolved work in only 3 rounds of sequencing instead of 9 that I have shown you before at the beginning. Not only that, you can take these technologies. They are highly versatile because you design what you want to do with this main street and back street. And if you are very smart, you can design them in a way that things can be, it grows exponentially, the type of things that you can do with those oligos. You can do thousands of cells, I have mentioned. We identified conformational characteristics. I haven't shown you anything about the biology we do with these images. I just brought to you the technologies that we have, and can be pipelined with other approaches, as I have mentioned, oligostorm, but also protein immunofluorescence, RNA, et cetera, we have seen beautiful examples in the previous talks. So I don't want to take more of your time. Just acknowledge who actually do the work. We are here talking to you, but who do the work. In this case, I have shown you the work of David Castillo and Irene Farabella, who were in the lab, but nowadays they have their own work. In fact, David works for this company, Acuity Spatial Genomics, which I also consulted for some time in previous years. Irene Farabella has her own group in Genova, and this original work is now being continued by Peter, John, Maria, and Mary Chey, who are here. So if you guys are interested in more details about this technology, just look for us, and we're happy to talk to you. Thank you.
Thanks so much. Questions? Yeah.
Hi, very cool. Really impressed by the technology. But something that I didn't quite understand, so let's say with the data that you showed on chromosome X, so you have very high resolution, but it looked like your the 2 copies of chromosome X were close together. Do you have the ability to distinguish, like when they're close in proximity, what the structure is?
Yeah, in that particular case, it's a male, so we only have 1 X. So what you saw, it was only 1 X. In the case of female, we have 2
or
things
any
that
other
are,
chromosome, right?
yeah, we have things that are advantageous to us, is that what is called homologous kissing is very rare. We still don't know exactly why in humans. We still don't know why 2 copies of the same chromosome tend to be farther apart than expected by random, except for the small chromosomes, such as 19, which are in the center, and it's much more difficult for them to spread each other. So this is an advantage. And then we have a version of this technology called HOPS, that if you have enough SNPs, then what you do is you design your oligos in a way that first you visualize the differential SNPs, so that I know 1 copy is here, the other copy is here, and then you do the rounds of sequencing to do the tracing of each one of them independently. And if they are close, you would still be able to differentiate which ones are belonging to here and which ones are belonging to there.
So for me, this is very curious that you have them at different portions of the nucleus, usually the 2 different copies of the chromosome, but you say this is human. In other species, it's not like this?
Well, for example, in fly, they are always pairing, and they are next to each other, and this becomes much more difficult to differentiate the pairs. And that would be the 2 extremes, for example. And there is anything in between, almost. One down here.
Is there any chance to do it for the mitochondrial genome?
Again?
Is there any chance to do this technology for the mitochondrial genome, mitochondrial DNA?
It's too tiny. It's too tiny to differentiate things. I can tell you we have tried high C, for example, and it's very difficult to see anything in chromosome conformation capture. We have not tried anything under the microscope, but it would be too small at this point.
Thank you.
It's not folded. It's not packed. Mitochondrial genome is not packed.
But you don't have to start differentiating things, you need to have enough DNA in between them to put the oligos, because you don't put only one oligo, you put thousands of oligos per signal. Otherwise, you don't see it under the microscope because you don't have enough fluorescence coming up from one oligo alone.
Yeah, well, I understand this is very recent technologies, but I see a very nice scope in relation to genetic variation. For example, when you show the two chromosomes, and you could see that one has a structure, or another structure, you say it is very plastic, but as well, I guess that genetic variation plays a role there. Is there anything already ongoing in that direction?
There is a lot of studies that look at variation at the genetic level compared to the structure. And I have to say that we know a few things. So for example, one of the most important transcription factors that regulate all of this is CTCF. And if you have variation on CTCF size, that means that you will start having variation in the structure because CTCF controls a lot of the structure. But we don't know enough yet to, for example, point mutations, what effect will have in the structure. This is one thing, so it's a difficult question. The other thing is that you have to imagine this is continuously dynamic. It's not a static picture. There is something called loop extrusion. This chromatin trace I have done for one cell, it's one particular moment in that cell. If I look at the same cell 5 minutes after, probably this structure is not there anymore. So it's very much dynamic, and there I think that the diversity in the structure is beyond the diversity that you can get by changing point mutations, for example. A different story are structural variants. Structural variants can have a huge effect, and that has been shown in disease, for example.
Thank you.
So what is with the quenching of the probes? Do they quench each other?
Yeah, they do, and if they are too close, then they quench each other. Yeah.
But when you say blinking, so that means you're actually going with very low intensity light, is that correct?
For the storm, you collect the individual blinkings stochastically. It's low. For the wide field, you cannot do that, and then you need all of them at once.
Yeah, but then you don't worry about the quenching.
Yeah. Okay.
Other questions? Well, thanks so much, Mark.
You're welcome.
All right. So thank you very much for the invitation. So today, I want to talk to you a little bit about some work we recently done using both single-cell RNA sequencing and spatial transcriptomics. So my lab is focused on understanding inflammatory bowel diseases, but and so I understand most of you are not interested in that, but I will use, because that's what I care about, the biology of these diseases, to show you some of the stuff that we did. But of course, hoping that showcase how you can combine these two technologies, which I think is really powerful. So inflammatory bowel diseases, very, very briefly, are a group of very heterogeneous diseases that affect the intestinal tract. And so there's actually a lot of heterogeneity, and we normally, patients will be divided into one of those two diseases. But for the sake of this talk, I'll just be talking about IBD, or inflammatory bowel disease in general. So they do have different characteristics. Disease locations, endoscopically and histologically, can be different. But in our lab, we try to understand what are the mechanisms, basically, why these diseases happen. And when they happen, how do they get complicated, more difficult to treat, more refractory? And we've been using a series of different approaches through the years to try to understand that. And for many years, as probably a lot of you, we used bulk RNA sequencing of intestinal biopsies. And that was extremely informative because we got, as you know, the whole genome transcriptome of these biopsies. But these biopsies are really complex tissues that include all different kinds of cells. And so to be able to disentangle which are the cells implicated in which step, certainly single-cell technologies have been tremendously helpful. And in the last 5 years or so, several papers, and I just highlighted some of them here. This is not at all exhaustive of all the literature in IBD and single-cell RNA-seq, but some of the ones that I think are most relevant. And through these new studies, we can now understand a little bit more first of all, what is the composition of the intestinal mucosa? Even though we thought we knew a lot about it, we've discovered that there were a lot of things we did not understand. For example, when you look at the fibroblast compartment in this intestine, it is way beyond what we thought could be there. Thanks to single cell, we now know. But what we're interested in my group is to look at what happens in patients, on the right side, when they have an active flare of inflammation and how this changes through the disease evolution, as I mentioned earlier. However, all of these studies that were published at the time did not have any multi-omic spatial analysis, and all the data was based on suspension of cells taken from these biopsies. So we started a project about 5 years ago. We started sequencing biopsies from these patients, and this was done here in Sinak with Holger, and we ran the first samples already in 2018, I believe. And this was done by 2 people in the lab, Alba Garrido, which is now a postdoc in Roche, and Ana Maria Corraliza, which is a senior bioinformatician in the lab. And the 2 of them took these biopsies, performed single cell here at Sinak, and then performed this analysis to try to understand the composition of the mucosa. And you can see here on the bars that this mucosa contains not only epithelial compartment, which is a major component of the mucosa, but also the stromal compartment, and a lot of immune cells. Every single immune cell you can think of is in the intestinal mucosa, basically. And so when we perform this analysis, then, and what I show you is a little bit of some of the biology that we learned from that, in particular in this study, and then how we then went into spatial transcriptomics to combine these and locate some of these populations that we identify. So, in this study, and this is what is published now, we focus, although we publish the entire data set and we talk a little bit about all the other cells, we focus particularly in the macrophage compartment. And this is because several reasons. One of them is the previous studies hadn't really looked into this compartment. And second, because one of the interests in my lab is to understand disease heterogeneity. And so why one patient behaves in a certain way and another patient that at the get-go looks pretty much identical to the first one ends up with a very severe, complicated, fistulizing, whatever condition. And so trying to understand this, and we look at the single cell data, we saw that where a lot of the diversity between different patients resided was among the myeloid compartment, and particularly in the macrophages. And we realized that there were different types of macrophages, which is well known in the literature. But by using single cell RNA-seq, we started to identify, I wouldn't say new types of macrophages, but different states in which macrophages can be found when we look at the IBD patients. And so in the healthy colon also, even in the healthy colon, we identify more diversity that was previously described. And we found that beyond the resident M2 macrophages, there were other macrophages there that were different from the M2. If you compare the gene expressed by both M2s and M0s, basically M0s have the backbone gene signature of a macrophage, but they lack all the M2 characteristic markers that have been described in the literature. We think that these macrophages, they seem to lack CD45 expression, and so we think that's maybe why we have missed them for so long, because we could not capture them using markers such as CD45 to begin with. But we did look into other data sets to see, can we see those M0s in others, and it's just not an artifact of our clustering, and indeed, we used the first 2 cohorts that were published in IBD, and we found that when we cluster this data on our own, the myeloid cells from those data sets, we could find cells in those data sets that resemble our M0s and also our M2s, and the same for this other data set. And furthermore, we did some stainings in the mucosa of the healthy colon to see, can we really see CD68 macrophages in green that do express the M2 markers, and some that don't. And as you can see here, there's abundant CD68 that lack expression of CD209, suggesting that this could represent more of the M0 population. But what was interesting to us is when we look in the IBD colon, there were several observations. One is that these M0s and M2s are not disappearing from the colon, because this is one of the things that we thought that, once you have inflammations, all the macrophages will become inflammatory macrophages, but we see abundant M0s and M2s that remain in IBD, although they do switch their signature. They adopt certain inflammatory genes. They do not lose their identity as M0s and M2s. But then, as you can see here, there's a lot of other macrophages, and also we found inflammatory monocytes that are well known to infiltrate the mucosa as any other inflamed tissues coming from the blood. But you can see here that there are several other macrophages. And when we look at their main markers, you can see here that we label them differently, and this is obviously just a label that we gave to them basically as M1s, more classically activated inflammatory macrophages. And this particular subset here, which was one of the focus of our study, these IDA macrophages. We call them IDA macrophages from inflammatory or inflammation-dependent alternative macrophages. We call them that way basically because we couldn't find a good marker. And second, because they're only found in the inflamed tissues, but they did not represent, they didn't have the full-blown M1 signature, so we couldn't really call them M1. So this is all this was nice, we can find these macrophages, but then we hit a wall, and the wall is we don't have good protein markers that we can use to really stain these cells, or more importantly, isolate them and then prove what is their function, etc. We tried, obviously, but we couldn't find any good protein marker that could be used. And a lot of these macrophages, we can just distinguish them based on a signature of genes that we can identify. And so this is when using a technology such as a spatial transcriptomics really help us. And so we had, thanks to our collaboration with Holger, he got access to this early access program from Nanostring, in which we were able to submit samples from our patients. And they performed the analysis. This is the CosMx 1000 gene panel. And then the data was sent back to us, and we performed this analysis that I'll be describing to you. So basically, very briefly, this was all done from paraffin-embedded tissues. The samples were cut there at Nanostring in Seattle, and they did the hybridization and the readout and the segmentation analysis. We actually performed 2 rounds of segmentation, and which was, the second one was improved over the first one. And then once the data was here in our hands, then we performed the analysis of this data. And what I want to emphasize here is that we use our single cell data annotation. So the markers that we had from our single cell RNA-seq, and then what we just do is this label transfer of these markers into the CosMx data set. And now, so this work was done by Elena Crowell, which is now a postdoc in Holger's lab. Initially, she was in Zurich at the time, so she performed all this analysis. And then Paula later on, when we had the second segmentation, she also performed this analysis with her. And now these analyses were continued also in my lab. So from this analysis, what you can see here in this UMAP is we got about, so these were 9 tissues from 9 individuals, including Crohn's disease, ulcerative colitis, and healthy colonic samples, and a total of about over 500,000 cells were analyzed. And here you can see that we were able to identify the different cell types like epithelial cells or the stroma, plasma cells, myeloid cells, etc. And here you have just the proportions across the 9 samples. And importantly, when Elena did this label transfer, what we could see is that we could indeed see all the different cell types within each one of these species. For example, if we look at the stromal compartment by single cell, we can identify at least 4 or 5 different types of fibroblasts, endothelial cells, pericytes, all those cells. Then we can label transfer and annotate here myofibroblasts, etc., or smooth muscle cells, I should say here. The same for the T cell and B cell compartment, for the plasma cells, the myeloid compartment, including all our macrophages, and the epithelial layer. And so, as I mentioned, our focus was the myeloid compartment. So I'm showing you here the markers based on the CosMx data analysis. And you can see that based on the CosMx, we can differentiate very clearly, for example, neutrophils here from mast cells that have these clear markers, or eosinophils also here have their own little markers here. And also within the macrophage compartment, you can see that there's certain distinction, although they do share, as you would expect, some of the signatures. We can differentiate clearly the M1 macrophages from the M0s here, or the M2s, or the IgA macrophages. So if we zoom in a little bit into the macrophage compartment, we confirm that in the healthy colon, these samples here, these are different fields of view or FOVs from the healthy samples. You can see here, maybe it's difficult to appreciate, but in red are the M2 macrophages, and then in light pink, we can see M0s here. So the most abundant macrophages in these samples were indeed M0s and M2s. But when we look at FOBs, samples from either ulcerative colitis patient or Crohn's disease, you can see that, first of all, there's a massive amount of macrophages. We can still see, as I mentioned, a lot of M2s, resident macrophages and M0s, but you can start to see a lot of IDA macrophages in green. And here, for example, this is an area that is an ulcer from an ulcerative colitis patient. Here, it's hard to see because this is very destructive tissue, but you can see here, this is the ulcer, the most outermost part of the mucosa, full of M1 macrophages. And then here you can see a lymphoid aggregate. This is it's hard to see, but this would be the submucosa here. So we could find these macrophages in the tissues based on those transcription signatures, so that was very important to us. But then we were very intrigued by the IDA macrophages. And as I mentioned, we didn't have a good marker for these macrophages that we could identify, although one of the markers that some macrophages in the IDA subset expressed is this one, NRG1, which is encoding for neuregulin 1, and it's a ligand for the EGF receptor. So it's known to act on epithelial cells and promote, their maintenance. So when we look, however, in the Cosmics data, what we found is that these IDA macrophages were highly abundant in these structures here. These are granuloma. Granuloma are these aggregates of macrophages that are characteristic of infections with things like mycobacteria, for example, things that are in intracellular bacteria, and try to contain the infections. They've been associated to Crohn's disease for a long time. We know for a long time that about 40% of patients with Crohn's disease will present granuloma, but the etiology of this granuloma in Crohn's disease is unknown. But it's characteristic, and one of our patients had abundant granulomas. You can see here that these were full with macrophages, and we were curious because a lot of these macrophages were identified by the Cosmics analysis in red as IDA macrophages. So we wondered, could we confirm this? Are these really IDA? Then they should express NRG1. But because NRG1, as I mentioned, is highly expressed by these macrophages. And also we know that NRG1 is highly upregulated when there's inflammation in Crohn's disease. This is bulk data. No good antibody for NRG1, of course, that we tested a ton of them, and none of them will be working well in our samples. So we had to use a probe and use in situ hybridization. In pink, you have NRG1, and in brown, you have the antibody for CD68. And you probably, I don't know if you can notice, but there are abundant NRG1 positive macrophages here. However, there are also NRG1 non-CD68s, and this is also shown in our single-cell data. There's a lot of fibroblasts that also produce NRG1, so that was to be expected. However, when we look in the granuloma, what we could see is that it is neuregulin. It is only found in this part of the mucosa, which is this upper part of the surface of the epithelium, close to the epithelium, which makes sense that the production of neuregulin, both fibroblasts and macrophages, are located here. But when we look in the granuloma, this granuloma did not contain, it really did not stain, or very little, for NRG1. So it was clear from that analysis, and this is what we published, because that's the data that we had, that we still have pretty much, is that these IDA macrophages are not just one single cell type, probably, or that the resolution we had with the Cosmic was not enough to identify and distinguish them from others. This is still a question that we are trying to ask. But the NRG1 high, based on the spatial transcriptomics, we could really see that they are located, NRG1 production is located to the upper part of the mucosa and close to the epithelium, but other identified as IDA macrophages or activated in some way do not express this and localize more within the granulomas. However, more recently, we have been able to increase our data set. So we have been continuing to sequence samples from patients. And out of that analysis, and this is analysis done by Angela and Mark, two PhD students in the lab, that have taken, Angela's taken this data set, which now includes more than 100 patients. The data I showed you was 18 samples. And clustered the macrophage compartment. And as you can see here, we still have these M0s, M2s, and then the M1s. You can see here expression, for example, CD68, which is all macrophages, CD209 for M2s, and then you start seeing here, for example, the CXCL5, only M1s, or ACOT1, also M1s, etc. But we also use in more cells, we find more clusters, as you would expect, and we found, for example, these clusters of macrophages here, these ones here, heat shock macrophages here, and also importantly, these IDA macrophages, they seem to be at least in two different states, IDA M1 and IDA 2. So, you know, I'm not trying to convince you of all this, the existence of all these macrophages, to see that this is a lot of diversity, and the more samples, the more data that we have, probably we're going to be able to find different things. But what I wanted to show you is that then using this new annotation, we repeated the label transfer into the CosMx data, and this is what Mark did in the last couple of months. And so when we do that, what happens is that we now have, in these same granulomas that I was showing you, we start to see other cell types, not just the M2s in purple, in dark purple, and the M1s, which are now very abundant in these granulomas, but also plenty of IDA, which are here in pink. Let me zoom in here a little bit. And you can see here that there's a ton of different states of the macrophages in the granuloma, with a predominant M1 and M2, but also IDA macrophages, but also the heat shock macrophages in green, these PLA2G macrophages, etc. Also, if you notice, certain macrophages, for example, these IDA that are closer to the M2, so they maybe be less activated, they tend to localize outside of the granuloma. So we don't find them so much inside, but yes, here, these red ones found in these other areas. Okay, so I'll would like to conclude just by saying that usually, obviously, we believe, and we have, you know, very privileged to have access to these single cell technologies from which we have learned a lot, not just my group, but the IBD community, but that we think that they really, all these single cell data sets, without this spatial resolution, it's very difficult to really understand the function, to really understand the heterogeneity, even within a cluster of cells. You can start, as I hope I show you, with this type of IDA macrophages, which represent different states. Having the additional spatial resolution really helps us to understand that heterogeneity. And also, one thing that I'm very interested in, and I know, you know, Holger and his group is also very interested in, is trying to understand how these cells interact to each other using the spatial transcriptomics, the receptor-ligand interactions, which so far we've been doing just from cells in suspension. Now we can do it in the tissue, so really having the coordinates of those cells and how that can impact the formation of different niches or neighborhoods and those interactions. So very exciting times, I think, for this area. Okay, and just want to finish thanking. I thank most people that were involved with the group, also all our collaborations. And I want to especially thank the IBD clinic, hospital clinic IBD unit, for obviously the access to all these tissues, samples, and all the insight into the clinics of these diseases, and of course all our funders and all of you for your attention. Thank you.
Okay, thank you very much, Azucena. Questions here? We have Jose. Yes. The microphone isn't on.
No.
Well, I think it's been very inspiring. I really appreciate all your explanations. And I wonder if all the heterogeneity associated to these diseases, to this group of diseases, do you think would be explained just by these inflammatory cells, or there would be other factors?
Definitely.
And then the second question would be, how do you feel when reading almost every week in the newspapers that nutrition is so important to, let's say, to cut off the symptoms of these diseases? Is it blah, 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 or is it something more?
Okay, two very different questions. So the first one, I don't think at all that this disease cannot be explained by one cell type. That's 100% sure. We know that the factors that can drive the development and the maintenance, which are could be, and it's proven at least from the genetic point of view to be different. So I think that obviously there's each cell type, but also the interplay between these cell types. So to be able to disentangle of this, extremely difficult, but that's kind of what we're trying to do. My focus on the macrophage was because we do find that if we look at the heterogeneity of the macrophage populations between patients, is where we see more differences in composition, in the states. We do the same with the epithelium, which changes dramatically during inflammation, or the fibroblasts, or any other cell type. And even though patients change, they seem to be doing it more homogeneously, in a way, okay? So that's why we think, and also other data that I didn't have time to show you, that we see the importance of these macrophages sensing the environment because they sense and adopt a signature. And we have evidence now that they sense also drugs and respond differently. So I think they're very cool cells, but certainly not the only ones. And regarding the second, I'm not an expert on nutrition, so I don't want to say anything. I think that it's obvious to everyone, you don't need to be an expert on nutrition, that the diet is going to influence a lot of things. Basically, it also influences the immune cells, but also the microbiota. But I think this is still very, very early ages to really link these two. Having said that, I think that there are many clinical trials now, controlled clinical trials, using nutrition to help, especially children, to help with the treatment of the disease and, maintenance of remission, which is very important, etc. But this is an area that is, so anything you read in the news, I think, has to be taken with a lot of, I would say just obviously you want to have a healthy diet, but other than that, there's no evidence.
Thank you, Azucena, for this wonderful talk. My question is, I think that the more technological advances we have, the more we understand about heterogeneity of the cells, even under healthy conditions in different tissues, tissue-resident macrophages and so on, and even more after in disease condition. So my question is, do you think that these different states, let's call it the states of the macrophages, can switch from one to the other, and do you see this during the evolution of the disease?
Yes, that's a very, very good question, Anna. Yes, and we have evidence we have a study that we did with patients undergoing treatment with a JAK inhibitor. So giving them a JAK inhibitor and then looking at them several weeks. And we do see, of course, you say, okay, they switch because, for example, patients that respond to treatment and go in remission, obviously you see a massive, okay? But what I think is interesting is these patients that do not respond to treatment. In those patients, what we see is a switch of the M1 macrophages to a state that is even more inflammatory. So even within the same populations, inflammation is still present, macrophages are switching. And then we also have a lot, we included in the paper, and we've doing some stuff in the lab, where you can do in vitro experiments and compare. And we can switch from the M2s, like MCSF, macrophages, you stimulate with certain stimuli, and you start seeing how they adopt what we see in the patients, the signature. So like if you have a signature of a macrophage that you have identified in vitro, you can then look, how is this, what does it look like? What type of, can I find that state in the tissues? And you can see that and understanding a little bit how they are transitioning. But yes, that's what makes it very interesting, I think.
Okay, final question. Yes.
So a little bit going to this in the same line, is there a particular transcription factor or family of transcription factors that drive these IDA macrophages? And
yeah, we don't know. I mean, this is one of the things that we're trying to understand. We've seen, and it's very early on, I don't want to say anything without more proof, but we have seen that you can activate M2 in vitro, for example, with MCSF, adopt an M2 profile. And then, for example, something like serotonin seems to be inducing some of the markers that we see in this. But for example, if you go with a full-blown activation, you lose that. It's kind of like in between states, but at the same time, they also express a lot of like good healing signature, like neuroregulin, but also a lot of EGF receptor ligands, VEGF, also some of them. But then when I say this, but then when you try to look at the real markers, sometimes they don't show up because I think there's a lot of heterogeneity. So we may have some that adopt this more of a wound healing and others that are more inflammatory. But we need more data to be able to say that.
Thank you.
Great. Thanks ever so much. Much appreciated.
Is there? Okay. Okay. I will be today having a role of being the introductory presentation for the following presentation. And I will start speaking about the long reads by speaking about the short reads. That, as you know, the short reads have made a huge impact in the last 20 years and in science, and they're increasing output and throughput and decreasing price, not made only their way into different places of science, but also in particular into diagnostics and reimbursed diagnostic tools. That is a little bit still a lagging phase for the long-read sequencing technologies, this diagnostic part, for all many different reasons. And that was a reason why the scientists have been trying to look for ways how to still use the short-read sequencers and still looking into these dark corners that are normally inaccessible with the short reads, using the synthetic long reads and the link reads. The synthetic long reads, just briefly, they are different from the other ones by covering to 6 to 10 kilobases only, but they are a little bit more continuous. You see this in the middle panel where they are compared to the link reads. And they are represented by today at least by two companies, the Element Bio and Illumina, while the linked reads span over really the full length of the molecules that you are working with, 200 to 300 kilobases. They are more dispersed. But for both of them, you have to sequence really very deep if you would like to achieve the same resolution as you do usually with these simple regions with the short read. So you have to sequence 4 to 6 times more. So it's not for free. And then come the comparison to the legacy long read. So what are their limitations and their advantages to these synthetic long and the link reads? Certainly everyone knows that the long reads, they have still limitations in the accuracy and with the throughput. It's quite expensive to buy the instrument, train the people, having auxiliary instrumentation and everything. But in particular in this case, the difference comes with the input material, that not only that is high molecular weight, DNA was in either of the cases, but it comes in micrograms in contrast with the synthetic long and link reads where it is in terms of nanograms. The long reads, the legacy long reads have the advantages again, as everyone knows. that you are sequencing the native molecule. And that comes with detection of the other bases, the modified bases and their phasing. And also you can base the variants, you can resolve very complex regions. And last but not least, you can detect variants in the highly polymorphic regions. One of the highly polymorphic regions is human mitochondria. And we have developed a method to sequence with legacy long read technologies, full-length native human mitochondrial genome. This method can be applied for either of the two legacy long-read sequencing technologies, whether PacBio or the Oxford Nanopore technologies. We in the Sinak, we have ONT, and so the proof of concept was done on the nanopore sequencing. Every one of you knows mitochondria. It is the powerhouse in almost all our cells, and it's being inherited by our from our biological mother. And the genome is the residue of its leftover from the symbiont, which used to be 2 million years ago a lone living organism. And the heteroplasmy in humans is very frequent. That comes during our life. As old we get more and more mutations, deletions. But in certain cases, this affects the functionality of the mitochondria. And then this can be classified into rare diseases termed as mitochondrial diseases. That led us to develop the method to enrich and sequence the human mitochondria, starting with genomic DNA. And each of the samples then is being digested with hexanuclease 5 that digests all the linear DNA in the sample, at least you hope. And therefore, because it's not perfect, you still have to do the dephosphorylation. Then comes crucial steps. Each sample is being split into few aliquots, so 2, 3, 4, depends on your experimental design. And you introduce the CRISPR-Cas technology, and in each aliquot, the guide RNA will be targeting different position of the mitochondrial genome. And that will be important later on in the analysis. Then you can everything pull together, all the samples, all the aliquots, prepare one sequencing library, and sequencing on the Oxford Nanopore technology GridION. Within few minutes, after you load the library on the GridION, on the screen starts to clearly show sharp peak at 16 kilobases, and that continues all through the run. And afterwards, you, of course, have to analyze the results that we also developed within this work, starting with the FASTQ files, finishing with the annotated VCF files, and we developed proprietary two softwares, the ONT demult and the Baldur. This is for demultiplexing, and this one is the Baldur is for variant calling. The demultiplexing works, as you can already imagine, about with the cut site position of the mitochondria. So in this case, in this example, the sample has been split in two aliquots, and in one aliquot it's being cut at position 3000. In another aliquot, it's at position 11,000. And after the sequencing, there are two, in this case, full-length reads. The read number 1, which will on the reference sequence be at position 3000, start and finish at the position 3000, and the read number 3, which starts and finishes at the position 11,000. The other reads, they start at the right place, but they don't make it to the position of the cat. So they are not full length, but they can be used, for example, in detection of the single nucleotide variants, but not for the deletions where you need only the full length. Full length, of course, doesn't mean the wild type. Full length means that it starts and finishes at the right place. It can contain still the deletions. So this is how well works the demultiplexing. It works 100%, this sort of demultiplexing. It's perfect, and it doesn't make any mistake. So you can safely pool all of your samples together while using the cut position. If you are using a high molecular weight DNA, like extracted from cell lines, you get from one gridiron flow cell tens of thousands full reads of mitochondrial DNA. That's of course different if you are sequencing clinical samples, because first of all, the samples haven't been extracted for this purpose, for long-read sequencing. And secondly, they have been sitting in the clinical lab fridge for many years, being the control samples. And so we received from Valdebron, from Elena Garcia, 15 clinical samples. 14 of them had, we knew, single nucleotide variant, but we didn't know the position, neither the heteroplasmy. We were able to detect all of them in a correct heteroplasmic percentage, and we were able to detect even heteroplasmy below 1%, which is apparently quite success in this field. We also were able to use various starting materials from biopsy, from oral mucosa or urine, so very uninvasive ways of biopsies. The different thing was about multiple deletions, which was the last sample, the sample number 15, where Elena told us that they are unable, with their approaches which are available currently in diagnostics, to correctly answer where the large deletion starts and where it's finished. What is shown here, even it looks like that you see, but it's that you see the end and the beginning, but it's actually all quite wobbly, and the correct position is not possible to determine. The same PCR reaction we put on the ONT, and there you see really where the deletions start and where they finish. Well, so why to bother to do some other method, right? So that was the answer to this question, is when we use our Cas9 enrichment of the mitochondria. We split the sample into 3 aliquots with the cut position at position 11,000, 5,000, and 3,000. And the first panel, or the circus plot, you see that you have only the wild type. And that's because this position is in the middle of the both deletions, so you cannot cut something what is not there. So you have only the wild types. While in the middle circus plot, the cut is in the middle of the large deletion, so you have the small deletion only and the wild type. And finally, at position 3,000, it is outside any of the deletions, and you see the large deletion, the small deletion, and the wild type. So this is a comparison between the long-range PCR passed on the ONT and the Cas9 passed on the ONT as well. You see that the same sample looks very differently, not only because of the 2 different colors. So the proportion of the reads is really heavily skewed by the PCR bias. While in the PCR, you think that you have 80% of the large deletion. It's actually present in reality only less than in 10%. You would think that you have almost no wild type, but the wild type is present in 10%, and the same you can see the difference is 21% of the smaller deletion is in fact 84% in reality in the sample. So this is a difference. What you can see, how what I wanted to stress out is the current state-of-the-art diagnostic approach, the long-range PCR being coupled with Illumina. That's how currently the people would have been diagnosed today using this method, and the method that we have developed in our laboratory. So we are able with this method to multiplex the samples without using any additional barcodes. Just by the cutting at different positions, we are able to detect single nucleotide variants in the heteroplasmy of below 1%, and we are able to detect and quantify correctly the presence of the multiple deletions in the sample. Well, these are the people who have been all working on this paper, and I would like to especially thanks to our informatics, to our friends in IRB and in the Waldebronn Hospital. So thank you very much.
We have questions? Yes, here, Jose.
Thank you for your talk. I really liked it. And I'm very impressed that you could detect heterogeneity and quantify heteroplasmy in mitochondrial DNA. Is this service available now? Do you offer it to other people?
Yes.
Huh?
As I said, this is an
I don't dare to ask how much is it, but thank you. It's a great advance, really. It was a pain in the neck to quantify heteroplasmy.
I hope that Rita will satisfy your curiosity what it can do actually beyond what I have presented from our paper.
Okay. Any other questions? I see no hands. Thank
Oh,
you
everyone
very
was
much.
so impressed.
And I am very happy that the technology improved and we can do much more for the patients. And I would like to show some of our genomic projects today, and mostly the ones when we are working in collaboration with people from CNAG. So mitochondrial diseases are clinically and genetically very heterogeneous, and the underlying causes can be in the mitochondrial DNA and in over 400 nuclear genes. This picture shows some of the most common phenotypes. There are children with infantile multisystem disease. There can be children with Lee syndrome or adults with mitochondrial myopathy or CPEO. These are the very typical presentations, but the majority of mitochondrial diseases present with many different ways, and usually with a combination of different symptoms. Previously, most patients had to undergo a muscle biopsy to find a diagnosis. However, in the last 10 years, exome sequencing and genome sequencing revolutionized the diagnosis of mitochondrial diseases. And in most cases, there is a first-line genomic testing. And because there are several copies of mitochondrial DNA in each cell, they have a very good coverage, and the heteroplasmy rate can be very well detected in genome sequencing and also sometimes in exome sequencing. There is a very large overlap between mitochondrial disease patients clinically and other neurometabolic or neuromuscular diseases. Therefore, also an unbiased exome or genome sequencing has a much higher diagnostic yield. In the UK, we have used whole genome sequencing to diagnose patients with suspected mitochondrial disease. Katrin Schoen is my geneticist colleague who did this study. And in more than 300 patients who previously were studied for common mitochondrial DNA variants, whole genome sequencing identified the molecular cause in about 33% of the cases. And interestingly, 62.5% of the new diagnoses were not a mitochondrial disease cause. This is also illustrating the large overlap between mitochondrial disease and other rare neurogenetic diseases. And there were some patients with a treatable disease in our cohort. There are just a few examples. But not in only our cohort, but many other rare disease cohorts, there is approximately 10% of the patients who have a treatable condition. And in other statistics, so other papers from an Australian group, they put this number up to 60% when there is a way to change the management of the patients based on the diagnosis. In the Solvardi project, also in collaboration with Sinak, we participated in the project called Treatabolome, which is aiming to highlight the potentially treatable variants at the time of the diagnosis. And first of all, we did some systematic reviews on some disease groups and highlighted treatable genes and variants. And the Sinak team identified set up the Treatabolome database, and it can be used now all over the world. So there are just two disease groups where we did these so-called Treatabolomes. One is on Charcot-Marie-Tooth hereditary neuropathies, and the other one on Lee syndrome. We also work on phenotype ontologies, and Tiloka Ratnaika is a pediatric fellow in Cambridge, and she set up a database called Mitophen, which is containing the phenotypic information of many patients with certain pathogenic variants in the mitochondrial DNA. So she selected 89 clearly pathogenic mitochondrial DNA variants and looked up almost 700 papers which were presenting the clinical symptoms of the patients. And she collected the clinical information of 6,688 individuals and put it up all into this database. And this is now available for everybody. And by comparing the phenotype of patients newly diagnosed with these variants with the patients who were reported, it is supporting the diagnosis of mitochondrial DNA mutations in larger genomic cohorts. So for example, in the Solvardi project with Ida Paramonov and Leslie Matalonga, together we are trying to analyze the mitochondrial DNA variants in this large data set. And we try to use the Mitophen database and the Mitophen score, which is a score illustrating how similar is a patient presentation to other patients reported previously in the literature to facilitate the diagnosis of mitochondrial disease. And now Tiloka is setting up the Mitophen 2 for nuclear mitochondrial DNA mutations. And in addition to facilitating the diagnosis of mtDNA variants, we can also use this database for many other, answering many other clinical questions. So for example, a new question, what we try to identify with these databases, whether sodium valproate, which is very well known to be a very toxic epilepsy drug in patients with polymerase gamma mutations, does it have any toxic effect in other mitochondrial disease? And this is an important question because valproic acid is a very easily available, very good epileptic drug, but currently no patients with any mitochondrial disease receives it because of the known toxicity in polymerase gamma mutations. So by using the Mitophen database, she showed that patients with POLG mutations who did not receive valproate had a much better prognosis. So it's clear that patients with polymerase gamma mutations should not receive valproate because that's significantly worsening their prognosis. But when she looked at patients with mtDNA variants, she could not find any case with a fatal or severe adverse event, and she could show that actually in patients with the H344 common mitochondrial variant, which is causing myoclonic epilepsy, the patients had a very good response for valproic acid. So hopefully it can be used in other mitochondrial disease. So the first-line genomic is still difficult to diagnose patients with a single mitochondrial DNA deletion. And there are two reasons for it. One is that in patients who are older children or adults with Kearns-Sayre syndrome or CPO, they do not have the deletion in blood DNA. Patients with a severe early onset phenotype, Wilson syndrome, they still have the single deletion in blood DNA, but it is selected out very rapidly in the first years of life. So therefore, the patients still need to undergo a muscle biopsy to detect the single deletion in adult age. However, sometimes in long-range PCR, even in adults, there was a very slight band which was highlighting that maybe in a very low level we could detect a deletion in blood DNA. And that would be a very relevant question because nowadays patients usually don't have a muscle biopsy. We do an exome or genome sequencing. So if we could also detect the single deletion, that would reduce the number of muscle biopsies. And in this project, we started a collaboration with Marta Gut, Ivo Gut, and Simon Heath when they used the Oxford nanopore technology to do the single molecule sequencing of the human mitochondrial genome. And this was exactly the patient who I showed you before with the very faint single band of the single deletion in a blood DNA. So they could detect very nicely in that patient the single mitochondrial DNA deletion even in the blood DNA. So it is hopeful that if we use it, we have to still validate it in several more patients, but maybe that will be a way to detect the deletion in blood DNA. And now I will talk about one other project which we used Oxford nanopore sequencing of the mitochondrial DNA for. And this project is when we studied an inflammatory myopathy called inclusion body myositis in collaboration with Felix Kleefeld, a neurologist in Berlin, Charité Berlin, and Werner Stenzel, a neuropathologist. I see that also my slides are a bit distorted, but I hope it won't disturb later on. So inclusion body myositis is a very common form of inflammatory myopathy in people above 50 years of age. This is often associated with some other autoimmune diseases, and the patients have a very common HLA-DRB haplotype. There are certain muscle groups which are commonly affected, and they are the finger and wrist flexors and the quadriceps muscles. They are the most affected muscle groups. And this disease has been researched for over 30 years, but the exact pathology and the molecular mechanism is still unclear. So you can see that when the patients are trying to make a fist, then the finger flexors are weak. There are the quadriceps muscles which are weak, and you can also see it in muscle MRI. There are three main histopathological hallmarks of inclusion body myositis. One is the endomysial inflammation. So there are CD8 positive lymphocytes, macrophages invading the muscle fibers. There is a very significant mitochondrial pathology. So these patients have a lot of regidred fibers, SDH positive, COX negative fibers in the muscle biopsy. And the third hallmark is the so-called rimmed vacuoles and autophagy. I started to work on this disease and understanding the mitochondrial pathology already in the 1990s, and this was actually my first international paper, and I did this study in the Montreal Neurological Institute in Eric Shoebridge's laboratory. And that time we showed that patients with IBM have multiple mitochondrial DNA deletions in muscle, and Eric beautifully showed that in each single muscle fiber, there was a clonal expansion of a different mitochondrial DNA deletion. In the meantime, other groups also showed that there is increase in multiple mitochondrial DNA deletions and duplications in the muscle DNA of patients with IBM. This study was done by whole genome sequencing. And there is not only IBM, where there are mitochondrial abnormalities from the inflammatory myopathy field, but there has been just recently reported that there is another inflammatory myopathy called polymyositis with mitochondrial pathology. We just call it PM-mito, where there are actually similar mitochondrial abnormalities like in IBM. And these patients have some non-specific muscle symptoms, muscle pain, proximal muscle weakness, but they don't have the localization of the muscle weakness. What I showed you is typical for IBM. There is some inflammation in these patients' muscle, but there are no rimmed vacuoles. And these patients respond to immunosuppression, while patients with IBM do not respond to immunosuppression. So Felix is working on a big inflammatory myopathy clinic in Berlin, and they followed up several patients with this so-called PM-mito and IBM at the clinic. And they managed to do follow-up muscle biopsies from 14 patients who originally started at PM-mito, but after several years, the second biopsy showed that they actually developed inclusion body myositis later on. And there were several mitochondrial abnormalities on the electron microscopy already in the PM-mito phase. So he came for 3 months to my laboratory this year, and he performed long-range PCR, and he showed that there are multiple mitochondrial DNA deletions in both PM-mito and IBM muscle DNA. And there is also some lower amount of mitochondrial DNA deletion in old age healthy controls. You can see that the increase with age. However, to better understand the localization and the heteroplasmy rate of this deletion, we sent the muscle DNA to Marta and Simon, and they performed the Oxford nanopore sequencing. And this was the picture of the single mitochondrial DNA deletion. And what they found in the patients with PM-mito and IBM, that there was an increase of several very different size of mtDNA deletions. And the heteroplasmy rate was ranging from 1 to 50%. In some muscle biopsies have more than other muscle biopsies. But that was a very significant change, both in PM-mito and IBM. And Simon even quantified the change, and in PM-mito, it was similar or even more amount of mitochondrial DNA deletions than in IBM. But why is it important to know where exactly the deletions are in the mitochondrial DNA? So there is an idea that if there are the piece of the mtDNA is missing, then at the break points they join again, and then it may produce a new protein. And so there are some bioinformatic pipelines to predict which newly generated mtDNA molecule will produce new open reading frames and new proteins. And because these proteins were not known to the individual, they were arising around age 40 or 50, they are predicted, some of them are predicted to produce immune response, and they may explain the inflammatory myopathy. So at the moment, in collaboration with Joanna Rohrbach's group in Karolinska Institute, we are trying to look into exactly, to study the exact sequences of these deleted mitochondrial DNA molecules to predict whether there is any new protein which could trigger the immune response. And also in parallel, we collect PBMCs from the blood of the same patients, and we are trying to compare this data to identify whether is it something which is really happening in mitochondrial disease. And it can have relevance for other mitochondrial diseases as well. And also recently, there is a lot of new literature coming up that maybe these deleted mitochondrial DNA pieces can be released from the mitochondria to the cell, or even from the cell to the outside the cell. And this can happen at viral infection, bacterial infection, aging, or different disease state. So we are also trying to understand whether it is happening in IBM. So Felix in my laboratory also studied the copy number of the mitochondrial DNA and could show that both in PM mito and IBM there is a reduced mitochondrial DNA copy number. And on Western blot, he could show that several subunits of the respiratory chain enzymes were reduced both in PM mito and IBM, confirming that the mitochondrial changes are very early present in the pathology of this disease. And ATP5A is a subunit which is usually not affected in other mitochondrial disease, like the single mitochondrial DNA deletion or other tRNA mutations. So in IBM and PM mito, we detected a very severe defect of ATP5A and also ATP5B, which is another subunit of the ATPase producing the ATP, were affected in this disease. So we asked the question whether is there a special role of ATP5A which can contribute to the pathology in IBM. So in a recent study, it was shown that in some viral infections, like Coxsackie virus or porcine flu, there is an increase in calcium. This study was done in heart muscle cells, and the increased calcium is activating some calcium-dependent proteases, and for example, calpain-1, and these proteases are going into the mitochondria and cleaving ATP5A. And the role of ATP5A cleavage by calpain-1 has been shown to trigger an NRLP3 inflammasome activation. So maybe this is somehow also linked to the IBM pathology. And it is shown that, in fact, in IBM, calpain-1 is cleaved. And there is still the question whether does it have anything to do with the formation of the mitochondrial DNA deletions, because the mitochondrial DNA replication is just happening under the cristae where the ATP5A is located. So to better understand what is ongoing in this PM mito and IBM muscle, we also did proteomics in collaboration with Andreas Roos in Dortmund. And we compared PM mito versus IBM, control versus PM mito, and control versus IBM muscle. And we did the volcano plots and proteomaps in all these comparisons. So I try to summarize what we found, I think, and what I think is relevant. So we confirmed in the proteomics also that in the IBM muscle, there was a significant increase of the calcium signaling, confirming the role of calcium signaling in this disease. And also there were very significant changes in ATP synthesis and oxidative phosphorylases. When we compared the PM mito with the IBM, then we detected some so-called IRES-dependent viral translational initiation, which means that this is a pathway what some viruses are using to translate their own genome in the host organism. So it may be reflecting a potential viral infection in this disease. And also in IBM, we detected increase in protein synthesis and degradation, which is a later phase of the disease, and this is associated with rim vacuoles and autophagy. So the new question is whether is there any role of some infection, some viral or bacterial infection in IBM? Because we still don't know what is causing IBM. It's definitely not a genetic disease because there is no families where more family members are affected. So there were also papers in the 80s and 90s when the methodology was not very good who suggested that there is a role of Coxsackie B or HIV virus in IBM. However, it could not have been proven to date. So we are trying to use also genomics, meta-genomics or meta-transcriptomics to identify a potential pathogens which could trigger these changes in IBM or PM mito in collaboration with Matti Kankainen at the University of Helsinki. So he's a geneticist, but his specialty is meta-transcriptomics, and he's trying to identify different pathogens in tissues in human diseases. So they developed a pipeline which is identifying more than 30,000 different known pathogens in any human samples. So they are trying to study now our patient's muscle, RNA-seq data from the patient muscle compared to control muscle RNA-seq to identify whether there is any trace of a pathogen in these muscle biopsies. So far, they could only compare the IBM with PM Mito, but they did not find two significant changes. But it's not a surprise because this is most likely the same disease group. So how can we explain what is going on in IBM? So we know that there is a mitochondrial defect early in the disease. There is an inflammation, and maybe there is some triggering infection. But how are they linked? So what we think at the moment that there may be some viral or other type of infection many years probably before the symptom onset, which is activating some calcium-dependent proteases, which may link to the cleavage of ATP5A, and maybe somewhere at this stage there is a mtDNA deletion formation or potentially mtDNA release, which would lead to the inflammasome activation and the inflammation. And this inflammation is then worsening the disease. And it's important to identify whether these are real pathways, because there are some drugs which would act on those pathways. So I hope that after we have some more results, we can answer these questions. And as a summary, so in this project, we could identify that there is a clear mitochondrial pathology in IBM PM Mito. There are mitochondrial DNA deletions, and there are changes in mitochondrial calcium metabolism early in the disease, and they are probably very important factors in the disease mechanism. We are trying to identify some infectious trigger, which may have happened several years before the disease onset, with also new genomic technologies. And also my question is also for what can we learn for other mitochondrial diseases, because patients with mitochondrial disease are very sensitive for infection, and maybe some of these mechanisms also contribute to this infection sensitivity in other mitochondrial diseases. And I would like to thank the collaborators from Sinak and from Berlin, and also many of my other international collaborators, and my funders and my group in Cambridge. And thank you for your attention.
Thank you very much, Rita. Do we have questions? So I think it was quite obvious that that with this deletion that happens in the IBM or in the PM Mito, the technique that Marta just showed is obviously very nice to sort of chase that up, because that's a really, really complicated structure to get at. And because they are single molecules, you can really sort of get the full concept of what is this going to make? What is going to happen if I have a sequence of this type here? And so one of the next things that we yesterday decided that we're going to chase up is getting the annotation mapped onto this, so that we can then say what the potential proteins are, or protein changes are. Good. Okay. Good. If there's no question, then we'll move on. Thank you very much.
Very much for everyone being here. And as he said, my talk is going to be a little bit different from what we have been hearing today, but I hope it's equally interesting. So I'm Margarita Barceló, and I come from the Mediterranean Institute from Advanced Studies. And let me introduce the main character of my talk. So that's my study species, the pearly razorfish. It's a fish species that inhabits these barren sandy bottoms of the Mediterranean and also the warm waters of the Atlantic coast. And they perform this curious behavior of burrowing in under the sand for protection and also for rest at night. So this species, besides being very cute, as you can see on the images, it's kind of like an iconic species in the Balearic Islands. So there we know it as a roe. And it's kind of like traditional, cultural, popular to go out and fish these species with family and friends. And it's, when something happens with it, it's widely broadcasted on the media, so it's something that it's very nice, at least for us, because when you try to do research, outreach, it's very easy because people want to hear more about it, so it's always a nice feeling to connect with people. And it also, as you can see, it goes very high in price in the markets when it's the season, so it's something that it's also important in economical terms. And because of what I've been saying, they suffer a very strong fishing pressure, and this fishing pressure has been managed by imposing a closed season during the reproduction of these species, and also with a back limit of 300 individuals per boat. This image that you see here, that's the first day of the open season, so that's when then the first day you are allowed, and all of the boats that you are seeing here on the image, or at least the ones that are standing still, they are fishing for these species. So you can imagine here being a population of these pearly razorfish in this area, and every single one of these boats is going to take up to 300 individuals. So you can imagine how for that population, this fishing pressure is very, very strong. In fact, my research group, they decided to study to see to what extent this pressure was, and they did, using underwater cameras, they counted the population density, comparing the exploited population and marine protected areas before and after this fishing event, and they were able to calculate that close to 60% of the population was removed from the area in just a week. So that was kind of like a starting point to decide to use this as a model species, because we can compare populations that are heavily harvested versus populations that inhabit marine protected areas. So there we started our research, and we have recently published some papers on basics on the biology of the species. So for this, we use this acoustic transmitter. So we put inside of the fish, we put like a small acoustic transmitter, and we were able to follow this population of fishes inhabiting one of these sandy patches, and the signal of this transmitter was received by this receiver. So we could monitor the movement and the behavior of these fishes. From this, we were able to establish the social structure of the population, where we could see that males protect large territories, that they are adjacent to each other, forming this kind of like a mosaic of territories one next to each other. And within these male territories, we find smaller territories of females. So they follow what is called the haremic structure, with one male and a few females inhabiting their habitat. So besides the social structure, we also studied the activity levels. So you can see here that it's the gray areas correspond to nighttime. So you can see that at nighttime there is barely no activity, and the activity starts right at sunrise. And because of this burrowing behavior, we could actually pinpoint at the time when they emerge from the sand, because the acoustic transmitters, they start emitting signals right away when they come out. So we can know exactly at what time each individual wakes up in the morning. And besides the experiments in the wild, we also took some of these individuals and we took them in these confined behavioral arenas, where we also measure their behavior. So on this example here, we just placed a new object within the arena of this animal, and we observe its behavior. So we have a measure of how bold they are when they are facing a new situation. In this case, how much do they approach to the object and interact with it. So with all of these behavioral experiments, we were able to actually characterize the individual variability on this behavior. So in the case of this boldness experiment that we did, we could separate individuals from being bold. Oh, wait, sorry, way too far. So we could characterize individuals being bold, so that those were the ones that they were interacting a lot with this toy, and individuals being shy or just like moving away from the toy and not interacting at all with it. And we did this experiment in consecutive days to the same individual to kind of measure the consistency of this behavior. And as you can see here in these repeated measurements, we have some individuals here that they are consistently more bold, and some individuals here that are consistently more shy. kind of like on the same line, we could do the same for the onset of activity, so that time that I said that they come out of the sand, and we can see how certain individuals, they start their activity right at sunrise, and other individuals here on this side, they start their activity over an hour after sunrise. And you can see also there is a preference for males starting their activity earlier than females. So why is all of this important? So this individual consistency in behavior, it will impose an individual vulnerability at being captured. So if you think about it, if we have a fisherman going fishing really early in the morning, all of these fishes that start their activity later, they are going to still be buried under the sand, so they will be less exposed to this fishing pressure, and also individuals that are less bold, are the ones that are more afraid of interacting with new objects, they might not go to the bait as easily. So we have kind of like a differential selection pressures affecting these different behavioral types. So we ran some simulations, and we could actually theoretically model that actually this would happen, that after many rounds of strong fishing pressure, the behavioral density will be very different in exploited population versus in marine protected areas, where in marine protected areas you would expect to have more individuals starting their activity earlier, and in exploited populations you will have individuals starting their activity late. So this kind of like selective, differentially selection has been very well studied in fisheries research, where we know that over the years of actually selecting for larger individuals, now fisheries have changed kind of like the size range of fishes, with, in general, now fishes being smaller than they were a few decades ago. So, and we know that this is based on size, so we were wondering what is happening with behavior. So if we know that there is a selection towards sizes, it could be that there is also a selection towards behavior. But the first thing we need to do to study this from this perspective is to actually go a little bit into the genetics of all of these behaviors. So what we did, we started to kind of think about this, and we realized that it would be very complicated to do any of this without the reference genome. So we found this project that was called the Catalan Initiative for the Biogenome Project, that they were giving funding for sequencing whole genomes from kind of like local interesting species, and we thought that our species was of local interest, mainly because of being these iconic species in the Balearic Islands. So we applied for the funding, and we got it, and we were able to work alongside Sinak to do our reference genome. So we did some short read sequencing and long read sequencing, and also cDNA sequencing. That they did their group of Marta in the lab, and that was great. And all of these went into the assembly and annotation team at the Sinak, and they actually were able to get at a very nice assembly at chromosome level that confirmed the number of chromosomes that it was already known by a karyotype experiment, but this way we confirm it at a sequence level, and we also got the genome size that is very close to other individuals of the same family, and we got a very good quality metrics in terms of assembly and annotation, and well, the genome just got published a few months ago, so that was really exciting. And I will give you now a little bit of overview of the things that I'm working in terms of like the genetic aspect of my project. But as I said, this was published just a few months ago, so it's just like a general idea of what I'm going to do, not that much in terms of results, but I hope it's interesting equally. So first of all, we were talking about exploring the individual vulnerability of these individuals and how this could be explored at a molecular level. So trying to understand what underlying mechanisms could be making certain individuals to have this preference for starting early in the morning or later at night. So for this, we did an RNA-seq experiment, and I don't have the results yet, but I can give you a little bit of the first thing that we have seen. And for now, we were able to identify certain genes that are related with the circadian rhythms in vertebrates. And we were able to see how these genes are actually expressed in a different way along the day. So we have like, this is individuals that we took the samples at different times, and you can see how this gene expression is increasing along the day, while these other 4 genes, their expression is decreasing throughout the day. So those are genes that follow negative regulatory feedback loop, where the expression of one of the genes is suppressing the expression of the other, and that becomes a cyclic expression and regulates the wake and rest of vertebrates. So we were happy to actually be able to identify all of these genes in our species, and to be able to actually determine that of course, it was going to be the same pattern, but at least to be able to find it in our species. So for now, that is where this is stage at, but we can continue and try to pinpoint a specific molecular mechanism, not at the circadian rhythm level, but more at this individual variability level. And in terms of population genomics, we contacted the Netherlands Institute of Ecology, where they have been working with a technique that it's called epiGBS, that is just reduced representation sequencing, where you cut the genome using a particular restriction enzyme that targets promoter regions of the genes, and we sequence these regions, but also we do some bisulfite conversion prior to sequencing, so we can actually get also methylation information on those. So we collected samples from 12 locations of these species, and some of them living in marine protected areas, and some other them living in fisheries. And the general idea is to try to compare genetic differences, genetic and methylation differences in individuals living in this, under these very different selection pressures. So for now, as I said, we just got the data back from the lab for this, but we were able to already find some population structure at the level of SNP data. So some of the populations they were separating, so that would suggest that there are certain genetic variants that could be associated with the origin of this population, and also in terms of level of protection. So there could be that there are certain genetic variants that are found in marine protected areas that are not found in fisheries, or the other way around. So we have to continue exploring this. This is just like the beginning. And then from the methylation data, we found something that it was expected, but you never know. So it's nice that you expect something and it actually happens. So we were able to see some variation in terms of age, where on this side, this axis actually goes in this way, in terms of like older individuals have a differential methylation than younger individuals. So I'm sorry, I don't have any final results, so that would be what we have so far, and we will continue exploring also in terms of not only population structure, but also in terms of genetic diversity, to try to see if actually that strong fishing pressure that I was talking about is also diminishing the genetic diversity in heavily harvested areas, to try to add a genetic perspective into the fisheries management, something that it might not be that well done today. So with this, I thank you all for your attention and all of the funding that I got for this project, and I will take any questions.
Yep. So, do we have questions?
Thanks, I really enjoyed your talk. So you're looking at the SNPs and so on in terms of the fishing, but the differences that you see in how early the animals wake up. So the gender differences, this probably cannot be determined by SNPs. I was wondering, so how is sex determined in these animals? Do you have anything like the XX, XY that we see in mammals, let's say? And then do you also see that BMAL1 or PER1, PER2, are they linked to the sex chromosomes? So can you explain that way why they wake up?
Well, the thing is, it's a sequential hermaphrodite. So they start they are first male, so they are first female, sorry, and when they reach a certain size, they become males. So the genome is the same in both sexes. So there should be no relationship in terms of SNPs related to the sex, because they will have the same genome throughout their lives. So that has to be something that is regulated at some gene regulation level, not at the gene sequence level.
And when you looked at this, did you look in the suprachiasmatic nucleus? Do they have a structure like this that drives the circadian rhythms, or where did you really?
Yeah,
look
that
at the
was
genes?
so. This first experiment was something like, let's see what we can do, because we have never done anything like this before. So we used the whole brain. So that would be also very interesting. So when I see all of these special transcriptomics, it's like, oh, I want to do that. Of course, I'm not at this point yet, but it would be very nice to see not only like how much is it there, but where, because maybe that could also be related with the, I think it makes more sense for this to be related with the preference rather than actually an abundance, because the abundance is going to be cyclic anywhere. So.
And then because of this curious sex kind of change that these undergo, do you think that there's like an evolutionary reason for the males to wake up earlier?
Yes, we think that it could be something related with territoriality. So if you have to defend your territory, probably the earlier you wake up, the more control you have everything. And also we have seen some kind of like harem chronotype, let's say, some way. So in the places where the males start their activity earlier, all of the females within his territory, they also start their activity earlier than other ones. So there are lots of factors of like many things going on there that it makes everything way more complicated. But yeah.
Very nice. So for the epigenetic analysis, which tissue do you take?
Well, for this, because we couldn't, we couldn't kill all of these individuals, so we were taking just the fin clip. So it was from the fin. So it's not as directly related to behavior. So it will be more like in terms of population genomics. But I don't know. There are some tissues that then you can look at. I know that in birds, blood methylation, it's related to brain expression. So then you can maybe just like doing maybe a preliminary experiment to look at how methylation correlates with expression in the brain, then you could. Yeah, but for now we only have fin.
If it's fin in all of them, it's okay.
Yeah.
Fin in multiple animals, but if you take blood from one fin from another.
Yeah, no, then it's a mess. No, it was and we try to also always use also the same fin, because you don't know. I mean, who knows what the difference are.
Any other questions? If not, then thank you very much.
Thank you.